We have a lecture on NIR spectroscopy for rapid screening of plant genetic resources. Our today's speaker is from biochemistry, and he is working in a division of germplasm evaluation at NVPGR New Delhi. He did his MSc in Govind Ballabh University of Technology, Pantnagar, and a PhD from IRI New Delhi. Sir, I request you for your elaborate introduction, because as we have discussed, sir.
Okay, friends.
How to you, sir?
Yeah. Okay, friends. So let me introduce myself. Is that myself, an agriculture graduate from Pantnagar University, and then did my master's and doctorate in biochemistry from IRI, New Delhi. And thereafter, I served in the Northeast India in Central Agriculture University at College of Horticulture and Forestry, Pasighat for 7 years. And in December 2009, I joined at ICR NVPGR. Since then, I'm working here. My area of interest is the biochemistry, that you can see the nutritional biochemistry or food chemistry component that I have been pursuing from my first job. And now, once I am at NVPGR, what we realize that we have to deal with thousands of samples. It's not in 100, they are in the thousands. And doing evaluation to such a large number through traditional methods is not feasible. So it is required, we should have some rapid methods which are accurate enough for at least for the purpose of screening. So for that purpose, we started working on the NIR spectroscopy, that is near infrared reflected spectroscopy. So so this is the main domain, you can say, at present for my work. So I will be presenting my talk on this topic. So today we will be discussing about how near infrared reflectance spectroscopy can be used in screening the germplasm, in identifying the trait-specific germplasm, and how it can be also helpful to the crop breeders for the purpose of doing nutritional breeding. So to begin with, I have some questions. And I would like to have a good participation. I would not like to talk to the computer. I would like to talk to my friends. So here is a question. Which variety, as per your assessment, are more nutritious? Old varieties, pre-green revolution, or the new varieties? And put us, when I'm saying this question, then put aside the specifically targeted variety, which are we call the biofortified varieties. Just leave aside the biofortified varieties and then answer this question. Hello? Anyone? I believe I'm not talking to computer. Unmute yourself, Anuj. Anuj, unmute yourself. Yeah.
Yes, sir.
Yeah.
I think it is old varieties.
Okay. And why you think so? There must
because
be
they are more.
Yeah. There must be some reason in your mind. Hello. Okay, I think we have lost Anuj. Anuj, you should soon join back. So it is a fact also. Here is some of the studies done over the period when the researchers compared the food composition data from the previous time and then the data which they are getting now. Old food composition tables and then the new food composition tables. The results were compared and it was found that there is definite decline in the nutrient concentration. And the purpose and the reason is very simple. One reason is that we have been doing crop breeding primarily for the food production to increase the production. The targeted was higher productivity. It was not a target to improve the nutritional profile. The target was to get the more production, more crop from same unit area. And another reason is that over the period, soil quality has also declined. The two factors together contributed to low nutrient density, and you will find that the new varieties are having more of these carbohydrate than the other nutrients. And another reason is also that consumers usually like starchy food, sugary and starchy food. If you talk of fruit, you will like to have it more sweeter. If you talk of grains, then you would like to have them more starch. So these are the reasons that over the period we have increased the product. You can say we have increased the production of starch and sugar compared to other nutrients. Is that clear? Hello? Okay, no response.
Sir, okay.
Okay,
So, okay,
Yes, sir,
sir.
it's okay.
okay, that's great.
We
That
will
will-
have,
Yes, sir. Yes, sir.
So, yeah, if you will keep sipping, that will keep my momentum high.
no, sir. We would have muted, sir. So by
Okay.
the time we unmute, it takes time.
Okay, that's
Thank you,
good.
sir.
Okay.
Okay, so can we solve the issues using the varieties in circulation? That's another question. Though the answer is that it is not possible to work with the varieties in circulation because these varieties are developed on a narrow base. Usually a small pool of varieties is used to develop the new varieties. So they don't have much variability. The diversity is not there in that. And the breeder prefers that because breeder does not want to introduce the wild characters, the traits which are not liked. So that's the reason that we cannot use the solely the popular variety can be one parent, but they cannot be the source for if we are targeting for higher nutrient productivity. Okay. So what is required that we are required to use the pre-breeding to introgress the genes, even the mutation breeding or the nowadays we are talking of genome editing. The purpose has to be that we are now required to target the nutrient productivity. Now government is also saying that we have already achieved the food security. We have sufficient food, but we have not achieved the nutrition security. So if we have to achieve the nutrition security, we have to look for the nutrient productivity. So I believe in near future, we can talk when the varieties are to be released that instead of talking of the total crop production, we can talk in the terms of protein production per unit area or iron and zinc production per unit area. So we can start talking of productivity in the terms of nutrients. So here's the example for protein that you can see that how much variability already exists for different traits in here we are talking of protein in different crops. Like in maize, you can see it is ranging from 5 to nearly 14%. Barley, nearly 7 to 18%. Chickpea, around 16 to 28%. In different crops, there is a big variability. So germplasm is a source which we can use to have a higher nutrient productivity. Now I'm taking it to another domain. Just think of, and from here onward, nearly 5 slides will be there, and you have to find out what is common in that. Okay. The assignment currently is that you have to find what is common in all the slides. Nearly 5 slides will come. Okay, so traits which are generally used in crop breeding. One is agromorphological traits like height, branch, root, shoot, architecture, pod, shape, size, color, days to flowering, days to maturity, and there could be different other traits. Similarly, resistance to biotic or abiotic stresses. Then here you can see, here you see the variability in shape of seed, pod length, pod color, seed color, crop maturity. Okay. Here again, you see the variability for different traits. These are some of the release variety of different vegetables from IRI. Here again, some varieties you see, release variety. And also in this. Okay. You have all seen the golden rice and the dwarf wheat and dwarf rice. Now you have to tell me what is common in all these slides. Anyone? What is common in all these slides? 1, 2, 3, 4, and 5, and then 6. Yes. What is common in all these slides?
Sir, pigmentation.
Nutrient,
Yes, pigmentation. But
nutrient,
yeah,
nutrient.
nutrient. Okay. I don't see mention of nutrient anywhere here on this slide, and on this side. And here also we are not talking of any nutrient here. Here, of course,
Sir, color.
color. Okay. Here, okay. Anything else? What is common?
Diversity.
Diversity. Okay. Anything else?
Size, shape,
Size, shape.
All are annual crop within.
Crop. Okay. 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 But what my target is to bring your attention is that these all traits are those which you can see through your naked eyes. Whether the plant height, root shape, architecture, seed shape, size, color, days to flowering, maturity, or even resistance to biotic or abiotic stress. You can see whether the disease is there or not. You can see whether the crop plant is affected by the abiotic stress or not. You can see the growth, right? So all these traits are visible to the eyes. Correct? But you cannot see the protein. You cannot see the starch. You cannot see the sugar. You cannot see the iron. You cannot see the folate. Can you see from your eyes and say that this grain will be having higher protein or this grain will be having higher folic acid concentration? Can you see that?
No, sir.
Yes.
No.
Yes. So that is the main difference. That is the reason that nutritional breeding has not progressed, because breeder cannot see these traits being inherited. Other traits breeder can work comfortably. They can see those traits. But how can they see that the which F2 or F3 population line is having higher protein? And the moment you give to the chemist to tell me the protein content, you lose the seed. Isn't it? Because let's say if you are talking of only protein, you will give the seed from the F3 or F4 population to the chemist. The chemist will do the digestion and then estimate the nitrogen and tell you the protein content. You got the protein content, but you lost the seed. Isn't it? But when you are talking of other traits, those traits were clearly visible to the eyes. You don't have to lose the seed. That's one reason that nutritional breeding has not progressed. So if you see in this slide on the right side corner, I have drawn a special eye. And by the side of it, which we are using, this is NIR spectroscope. So this NIR spectroscope acts as a special eye, and special eye which can see and tell us how much the protein is, how much the particular amino acid is, how much the phenol content or folic acid is there. So all these things are possible to be estimated without losing the seed. So that is the reason we prefer to work through NIR. So then it is non-destructive, rapid. It does not require use of chemicals. So it is not hazardous to environment. And moreover, if you do chemistry and you have to work on the thousands of samples, you will be required to engage at least 3-4% to do the analysis for a single trait. Now there will be a variation in handling from one person to another person. Then there is a possibility of error at every step of estimation, from the time of sampling to sample weighing, to sample homogenization, to sample extraction, and then to sample estimation. Every step there is a hazard of error. There is a chances of error. But in NIR spectroscopy, after sample is taken, it is homogenized and then kept in cuvette. Then it is scanned. After that, there is no chance of error. Whatever error is possible is that if you can do error in sampling, or inaccurate homogenization, or improper packing in cuvette. If just these three things, you do the sampling properly, you homogenize and sieve it properly, and then pack it properly in the cuvette. After that, there is no chance of error. So whatever error is possible only at these three steps. But if you are doing wet chemistry, at every step there is chance of error. So doing chemistry for thousands of samples is not only expensive, but also the error prone. There will be more error in doing estimation by actual chemistry compared to when using the NIR spectroscopy methods. Any queries up till now?
No, sir.
Okay. So NIR spectroscopy is used in different domains, like in drugs and pharma, in soil chemistry, in dairy and beverage industry, in food chemistry for estimation of different nutrients in seed, flowers, fruits, and vegetables. Even for different kinds of food sources. Even the processed food is also can be estimated. Now, it can be used in two ways. One is when we develop the trait-specific models and use to estimate them. That it comes in the domain of supervised learning. That what we do, we take the spectra of each sample, and then we take the chemistry data for each sample. And then using the chemistry data and the spectral data, we develop the prediction models and use it to screen the germplasm. When we screen the germplasm, after screening the germplasm, we can identify the trait-specific germplasm. Then those trait-specific germplasms can be used in nutritional breeding. And then again, this NIR spectroscopy can be used to study the inheritance of those traits. That's one domain. Another domain is unsupervised learning, where we will not do any chemical analysis. We'll simply scan the sample and then cluster the similar types. You would have seen developing the hierarchical clusters, or PC1 and PC2 plots, principal component 1, principal component 2 plots, principal component 2 and principal component 3 plots, different combinations of plots. So there what we do, we group the similar types, and that way we can identify the off types, and or you can say even the unique types. The unique types may fall in the off types category. Besides that, this can also be used to develop the biochemical course, because NIR spectra is influenced by the biochemical composition. So when we are grouping the accessions based on the NIR spectra data and then take the sample from each group. Actually, we are selecting the biochemical core. So let's talk something about the spectra and the NIR spectra. So you can see that spectra is very wide, starting from gamma rays and ending towards the radio waves. And the visible region is a very small region. Very small region is the visible region. And to the right side, or you can say towards the lower energy, is the near infrared. And near infrared is the subset of infrared region. So near infrared region is the region which is close to the visible region, or you can say starting from the boundary of visible region. And thereafter, after near infrared region comes the mid infrared region, MIR, and then comes the far IR. So in this, IR is used more in the pharma industry because it gives the fundamental vibrations, or you can say it gives the specific signature of a particular compound. So IR is used more in the pharma industry, although in food industry is also used and can be used. But near infrared region, although it looks, if you see, okay, we'll come to that. Near infrared region gives the combinations, overtones and combinations. So its spectra is more complex, but it reveals the more complex information also. What happens when the near infrared light passes through the sample? Then wherever there is a polarity in the organic compound, which is due to dipole moment between the polar and highly electronegative molecule and carbon, like carbon and oxygen. So oxygen is electronegative, it will pull the electron more towards it. So there will be dipole moment, or you can say polarity. So there it will cause stretching or bending. So there is a stretching or bending in the functional groups that lead to the loss of energy. And then those energies released in the different wavelength region. So gain in energy in another region and loss in energy in another one region. So as a result, spectra is altered. There could be different kind of stretching, like symmetrical stretching, that the both atoms are stretching in the same direction or in the opposite direction. There could be scissoring or rocking kind of movement, or wagging or twisting kind of movement. So here you get this kind of spectra you get. And now you see that this spectra is very sloppy. You will not find much sharp peaks. It's zero order spectra. But once you derivatize this spectra, you will see in the second derivative or in the third derivative, they are the sharp peaks also. So that way you can reveal some information. So so the spectra is treated to higher order derivatives to get more signal to noise ratio. They are the different processing methods. Now coming, how any questions up till now? Hello.
No, sir. Continue.
No, sir.
Okay. So if we have to develop the prediction model, we prefer that set of more than 100 sample. But although if the good diversity is there, even in the 40 to 50 sample also, one can develop a really good model. But the requirement is how much diversity is available. So diverse sample set, then NIR spectra is required, and wet chemistry is done on the same sample set to generate the data, which then this data is act as a reference data which is used to train the computer. Now this diverse set is split into two sets. One set is training set or calibration set, another set is testing set or validation set. After that, what we do the spectra normalization, like you saw here previously that the spectra is derivatized. Likewise, derivatization, smoothing, scatter corrections, they are the different normalization methods which are applied. And if there could be some outliers, then those outliers are removed. Then regression methods are used. They are the different types of machine learning methods. Most commonly used is the partial least square regression methods, PLS methods. But where you don't get the linear relationship, then we are required to use the machine learning methods like support vector machines, artificial neural networks, or deep learning methods, or ensemble methods. So those methods are used. Then prediction is done on the validation set. So remember, they are the two sets, one set on which the computer is trained, and the another set, then the developed model is used. It cannot be the same set. If you will be using the same set, then the result can be error prone. So it is always good to have two sets, one set for training, another set for testing. And then the best fit model is selected. Now, if you have to select the model, we follow that R squared, that is called power of determination, should be more than equal to 0.8. And then standard deviation to standard error of prediction ratio should be more than 2. If it is more than 2, it qualifies for screening criteria. So there it will pick up the general trend, but it may not be precise. But if it is 3, then it is considered good. And if it is 4, then it is considered equivalent to wet chemistry. So the data which you will get from wet chemistry and the data which you will get from the NIR prediction, they will be equivalent. And if it is more than 4, it is considered better than wet chemistry, because we know wet chemistry is having the possibility of error at each and every step, whereas NIR prediction model, there are not much possibility of errors. Here we are presenting you the chickpea NIR prediction model for protein and starch. You can see how much good correlation is there between the NIR prediction value and the actual chemistry value. They are in much good agreement. You can see that the laboratory value and predicted values are falling on close to the straight line, and slope is also close to 1. Here is another example we are presenting for the chickpea only. You know that the sum of proximate is the international standard, that the sum of proximate should be 100 plus minus 10. That is, if you are estimating each and every component of proximate composition by chemistry, then the sum should lie between 90 to 110. And here all the proximates are predicted, and the sum of that is also again within the range of 100 plus minus 10. So that way, this is also an indirect proof that NIR prediction model is working very in a very good way. You can see that here blue line is for dietary fiber. Where dietary fiber is low, you can find that the starch content is high, and inverse relationship is there. So at NBPGR, we have developed the NIR prediction model for different traits and different crops, and we have been using it for screening the germplasm. You can see the different crops like amaranth buckwheat, rapeseed, mustard, safflower, soybean, chickpea, eggplant fruits, rice bean, adzuki bean, French bean, different legumes, multiple legumes, then rice, pearlnut. Nowadays, we are also working on potato. We have developed the model for turmeric also, for curcuminoids. So how it is used now? So these are some of the publications from our laboratory where we have developed the NIR prediction model in the recent years. Okay, now coming to the usage. So as we have the good NIR prediction model for rice, we use it to screen 395 accession of rice land races from Assam. And based on that, we could identify trait-specific accession for high protein, accession for high oil content, for low phytate content, for high phenol, and rare combination also we could identify for high amylose with high oil. It's a rare combination. Accession with high amylose along with high oil. It came true. And the advantage of high amylose with high oil are two. One is in the cooking property, it gives the springiness to the cooked grain. Another is its low glycemic index. The glycemic index is reduced because amylose is a straight chain, so it takes time to digest, whereas amylopectin is branched, so it gets digested very quickly. So if the amylose content is high, and along with oil content is also high, you know that enzymes are proteinaceous in nature and they are hydrophilic. So if inherent oil content is high, then that oil acts as barrier to the enzyme to access the amylose. As a result, digestion rate is slowed down. There are some of the findings of the unique rice land races with high amylose, with low amylose, with medium amylose, with high oil content, or with high protein content. And then amylose to amylopectin ratio. So based on that, our study we targeted 17 accessions. Which could have, hello. Hello. So based on that, we targeted, based on the prediction value, we targeted that these 17 accessions have a possibility of having low glycemic index. So we gave those samples to the rice laboratory in Division of Genetics IRI, and they estimated it. And when I asked about the result, they said that, sir, what is that? That all samples are coming low glycemic. And they were doubting their results, that there must be something wrong must be happening. Although they were following the method very carefully, but when all the samples were giving low glycemic value, they started doubting their results. I said that, okay, I give you the another sample and I gave the high, which I suspected to be high glycemic. And I gave that sample to them. They said, no, sir, that sample result is good. And then the previous sample result is also good. They were bound to be low glycemic. And the reason was simple. These are the samples which are having high dietary fiber, high amylose, high phytate, and high oil content. If this combination is there, they were bound to be low glycemic. And you can see we could found even the estimated glycemic index of 44, and less than 55 is low glycemic in the rice. So the landrace Lotus Ali is having estimated glycemic index of 44. Very good for the diabetic person. Okay. So any question up till now? Hello? Any questions?
No, sir.
Correct.
No, sir.
Okay. So now coming to the another usage. I started talking of how we can help the crop breeders through the NIR spectroscopy. Here's the example where maize kernel single grain models are developed for the maize kernel. Okay. So if we have a single grain model, up till now, we at our laboratory, we have worked only for the flowers, using flowers. So you can say it is destructive from the germplasm point of view or from the breeder's point of view. But from the chemist point of view or from the food point of view, it is non-destructive. So we have to start work on the developing models for the grain, single grain. So if we have a model for the single grain, then those model can be used in assisting the crop breeders. Besides that, these NIR spectroscopy is also used to predict the seed germination, viability, and vigor in soybean, in pepper, in different crops. Now, from the gene bank point of view or from the seed industry point of view, both ways, it is important to have each and every seed should be viable. If we are storing any seed which is non-viable, it is loss because it involves a lot of investment. So by developing a model for seed germination, viability, and vigor, we can have a system where we can trigger out all the non-viable seeds and ensure that what we are conserving is 100% viable seeds. There's again some publication where the seed viability and vigor is being predicted using the NIR spectroscopy. It is also used in the, just for your information, estimation of soil properties, soil chemistry, like what is the soil pH, soil organic carbon status, soil nitrogen status, all these things can be predicted. Then we talk about geographic index, GI. Okay. And we have the GI released in the different crops. Now, how to ensure that the customer is getting the material which is true GI, or the person who is in the marketing chain ensures that he is getting the right material for supply. Here also NIR can help. NIR spectroscopy can be used to identify the regional variations. The same variety grown in different environment have different composition. Their composition is influenced because of environment. The basmati rice which is cultivated in Dehradun will not give the same level of aroma and same kind of composition, the same variety which is cultivated in Delhi. Okay. So GIs can also be, and for that unsupervised machine learning or clustering is required. You don't need to do any chemical analysis. Here's the example of identification of mutants. So if you see in this, the samples which are coming within this circle, they will be the general population. They will be non-mutant, because once you do the mutation and then have T1, T2, T3 populations, many of the mutations revert back. Only few mutations sustain. So the fourth quadrant, or you get the right side upper layer section quadrant, in that which are coming outside the boundary, they are the definite mutants. And the one which is on the right hand side, the lower part, outside the circle, they are the possible mutants. But the one which are on the upper part, outside, then these are the definite mutants. Or you can say definite outliers. So mutants can be usually classified. There are some other studies. So NIR spectroscopy has been used to identify the and classify the mutants and distinguish the mutants from the non-mutants. Then here's another example of using the NI spectroscopy to classify the botanical origin and geographical origin of honey. So the honey is coming from a particular specific flower, botanical origin, or from particular region. So for that purpose also NI spectroscopy is used. So one can distinguish the GI also, one can distinguish the mutants, one can identify the off types. One beautiful example we got during our study, we scanned nearly 6,000 accessions of barley. In fact, they are flour. Okay. And then when we clustered them, only one accession was there, which was unique and which it was falling in the separate cluster. Single accession. We got interested in that. By composition also it was unique. But when the field grow test was done, it was identified it was not barley, it was wheat. And it was a wheat which was having tight husk attached to it. So because the husk was attached, so it was misidentified when the explorer collected, because the explorer found that the husk was attached to the grain, so it recorded as barley. It was there as a barley. We estimated, we simply did the NI spectroscopy, it fall in the separate cluster. When we did estimation, it was unique in its composition. When we grew it, it came out to be wheat. So out of 6,000 accessions, there was only one wheat, and NI spectroscopy which could identify that it is not the barley. So there are some of the areas where we can use the NI spectroscopy, such as development of biochemical cores without doing any chemical analysis. Identification of biochemically diverse and distant lines for use as parents. Identification of mutants in mutation breeding. Development of trait-specific reference set based on prediction and validation. Non-destructive phenotyping of breeding population for nutrients and study trait inheritance. Distinction of land races and varieties, or regional distinction, or you can say geographic identification or GI validation. And selection of fully viable seed for conservation in gene bank. So this is all from me. Now I welcome your questions.
Thank you, sir. Now I request all the participants, if you have any question, you can ask. Now you interact with sir. Any question from your side?
Any breeder in the group?
Any question, please ask. Any question, please? If don't want to ask, you can also type a question. Anyone, please.
Or should I start asking by name?
Hello, sir.
Yes,
Hello, sir.
Sasikanth.
Sir, what can be taken for handling of NIR machines?
Yes, I missed the question.
Again, you ask, please.
Sir, what care should be taken for the handling of the NIR machines?
Okay. You should maintain the instrument in clean environment. Voltage fluctuations as far as should be avoided, which is for any instruments. And maintain the temperature of the room. But that's for the machine part of it. It should be kept clean. But the important part from the sample handling is that the sample should be homogenized properly. We use the cyclotech milk at the which we place the sieve of 1 mm. So any sample when it is grinded, it is passed through 1 mm sieve. So we get the homogeneous particle size. So and then once you fill the cuvette, just tap it gently and then scan it. And if you are doing with whole grain, then just also ensure that sample is clean. It is not having chaff. And moisture content, you prefer to use the ambient moisture. The sample should be first dried properly and then let it cool down and regain some moisture from the environment. Let it get stabilized. That is better. Instead of drying and keeping in desiccator to ensure that the sample is 100% dried, that doesn't work. Because by the time you will take out the sample and will doing scanning, it will start gaining the moisture. So better the sample is stabilized.
Any other question, please?
Hello? Hello?
Yeah.
Yes.
Sir, how much cost for this equipment?
Depends. If you have to purchase the research grade instrument, it will cost around 40 lakh.
Right, sir.
But there are instrument if one can get in 25 to 30 lakh also.
Thank you very much, sir. Thank you very much, sir.
Any other? I think, sir, there are no question now.
Okay.
Okay, then, sir, one another guy. One more question.
Visual infrared,
Dr. Vinod.
which is more reliable result.
Yes, yes.
For reliability of result, 2-3 things we have to consider. First is that the model which is developed, how much diversity is there on the sample used for developing that model. For example, there are very good models developed by the instrument industry. Like the one which we are using, FOSS. But the model developed by FOSS are of no use for us. Because those models are developed on the market sample. And in market sample, you will get the sample primarily of varieties. And we know that varieties are developed on the narrow gene pool. So there is not much diversity. Although these models are developed on thousands of samples, not on hundreds, because this industry take this sample spectra and chemistry data from around all over the world. So the model developed by them are robust, but they are of use for the market sample, not for the germplasm. This has been the experience even at the ICRI site. They were using the model provided to them by the FOSS. This is one of the very old and renowned NIR instrument manufacturer. And they found that the model was not able to correctly estimate the extreme values. Model was a big failure for the extremely high or extremely low values, because the model was developed on the narrow base. So first thing is that how much range of diversity is used to develop the model, instead of how many samples which are used to develop the model. You can use thousands of samples, but if the diversity range is very narrow, it will work only for the narrow range, because computer is like a kindergarten student. Whichever book you have taught him, it will answer only for those books. You have not trained the computer for the 30% protein. If the computer is trained for the 20 to 25% protein, neither it will estimate correctly the 15% protein nor the 30% protein. It will fail, because it has got training only to 20 to 25% protein. So first thing is that how much range of diversity is used to train the computer. Then once you have done the validation, that how much good correlation it is giving in the validation, that is the set which is not used for training, and how much less is the standard error. And then usage of that model to another set, what kind of result you are getting. Okay.
There is another question, sir, here.
Yeah.
Machine will differentiate the sample on the basis of organic produce or chemical produce.
See, organic compounds which are having some polarity, some dipole moment, NIR works for it. So organic compounds are also the chemical compounds. So it sees the overall variation in the chemical composition, which could be because of organic compounds and as well as organic chemistry, or you can say synthetic compounds also. That's why NIR spectroscopy can also be used to check the adulteration in the food products. Like in spices, colors are used, like in turmeric, yellow color can be added, or in red chili powder, red color can be added, or in honey, fructose syrup is added. So all those adulteration can also be identified, not only identified, but also quantified by using the NIR spectroscopy, provided we train the machine.
Okay, thank you, sir. Any other question? Okay, then, sir, I may propose a vote of thanks, sir, formal vote of thanks.
Okay.
I, on my behalf and on behalf of organizers, I thank you very much, sir, for such a wonderful lecture you have delivered on NIR spectroscopy for rapid screening of plant genetic resources. Sir, you beautifully explained the estimation of different parameters of protein, like protein, sugar, oil content, etc. Large number of sample using this near infrared reflectance spectroscopy. Sir, you have given time in your busy schedule.
So
We are very much thankful for you, sir.
It's my love.
Thank you very much, sir.
Okay. Okay.
Okay, sir. Now we will have another lecture at 10:45. So be online. Our next speaker will join soon. You can have a tea in between. We have time. Sir, good morning. Huda sir, good morning.
Good morning, Subhash. Good morning. Very good morning.
Okay.
So, can you tell the today's lecture schedule?
Sir, yes, sir. Sir, today's next lecture is pre-breeding and wide hybrid.
Yes, sir. So, sir, we welcome you in this training session. And just for information, this training has been, organized on the request of ICR. And we are organizing this in collaboration with HRM unit of ICR, because ICR DG has mandated that NBPGR should organize these kind of training regularly on PGR awareness and management. So with this, we welcome you, and I would like to take opportunity to introduce our speaker of today, Dr. Aditya Pratap. I'm just sharing the introduction slide. Is this visible?
Yes, sir.
Okay. So friends, this is just for information. Dr. Aditya Pratap, born on October 18, 1976, and he is currently working as principal scientist in the Crop Improvement Division at ICR, Indian Institute of Pulses Research, Kanpur. And also presently he is working as in charge project coordinator, AICRP program on chickpea. He is holding a brilliant academic and service record. He has worked on genetic improvement of crop plants, including Vigna species, wheat, triticale, rapeseed mustard, and chickpea. He has been associated with the development and release of 14 crop varieties, including 9 in green gram, 1 in chickpea, 3 in rapeseed mustard, and 1 in facultative winter wheat. He had led the development of world's first earliest variety of mung bean, that is known as Virat, which matures in 52 to 55 days, besides Samriddhi through marker-assisted approach. He has also executed several national and international collaborative projects as a team leader, and successfully established the International Mung Bean Improvement Network, along with several international partners. To his credit, he has published more than 220 research papers in several peer-reviewed journals. He has edited 6 books published by Springer, Cabby, and elsewhere. His research interests include distance hybridization, double haplide breeding, plant tissue culture, and molecular breeding. He is a recipient of prestigious Lal Bahadur Shastri Outstanding Young Scientist Award and the Norman E. Borlaug International Agriculture Science and Technology Fellowship, USA. The Mung Bean Improvement Program led by him at the ICR IIPR has won him the Best Center Award in 2018. He is on board several reputed international research journals, besides the current General Secretary of the Indian Society of Pulse Research and Development. And friends, this is also, I would like to inform you, he has organized an international conference on pulses that has just, summarized on 12th of February. That was very nice, organized, and very well appreciated by the audience and stalwarts of the pulses. So with this, I would like to request Dr. Aditya Pratap for a presentation. And I hope our participants will enjoy the presentation. Thank you, sir.
Thank you, Dr. Kuldeep, for such an elaborate introduction. It could have been short also. Thanks a lot to you. I can not read all the names, but with my regards to all the participants in this conference, Dr. Kuldeep, who is here, Dr. Jyoti, I can see, Dr. Subhash Chandra is there from NBPGR, and my dear, Dr. Padamja is there, Dr. Niragundi. So a very good morning to all of you, all the participants. Today, as I have been assigned the topic, I'll be sharing my presentation with you. 1 minute. So you can see my slides?
Yeah.
Yes, sir, visible.
Okay, fine.
Okay. Thank you. Okay. So the topic I will be dealing today will be pre-breeding and wide hybridization, role of plant genetic resources. And I will be focusing on different aspects, including first the concept of pre-breeding and distant hybridization, then a need of decision making. Actually, this is one of the most important steps while we are resorting to pre-breeding and distant hybridization. Then uses an introgression scheme from different gene pools, because we have primary, secondary, and tertiary gene pools, and an introgression scheme requires a focus on what method has to be adopted for introgression. Then the presentation is slowly moved to pulses, because I'm citing a few examples in pulses and then specifically within a species. Then we will go for plant genetic resources, food and agriculture, about their conservation and their evaluation, and then how they can be used in distant hybridization. Then I will be discussing some pre-breeding efforts and successful examples, and then future thirst and conclusions. The first thing first, actually, when we resort to pre-breeding, basically there's a very thin line between breeding and pre-breeding. And when we think about resorting to distant hybridization and pre-breeding using the members from the different gene pools, especially secondary and tertiary gene pool, we need to be very clear about what is breeding and what is pre-breeding. Actually, as far as breeding is concerned, and most of us are focusing on breeding activities. So these are the activities which are designed to change the traits of domesticated plants in order to yield finished products. So finished products means basically it is the varieties. So whatever breeding we are doing for a certain objective of development of improved varieties, be it for yield, be it for yield-related traits, be it for quality, be it for maturity duration or other traits. So the ultimate aim in breeding is to get a product. However, in case of pre-breeding, this is not the aim. The aim here is entirely different, and it is to develop intermediate set of materials so that those materials can be deployed in breeding. So pre-breeding is basically this is activities designed to identify desirable traits or genes from non-adaptive materials and transfer these traits into an intermediate set of materials. Because ultimately, whatever materials we are developing through pre-breeding, they have to find a destination, and that destination is ultimately culminating into development of finished products by way of breeding. So obviously, if we are starting with pre-breeding, then gradually we have to shift to breeding, and ultimately we have to go for development of products. We can directly relate it with time also. For example, a breeding cycle typically takes 6 to 10 years, depending upon the kind of crop we are dealing with. Whereas if we start with pre-breeding, this entire cycle, it may take around 15 to 20 years for successful completion. As far as plant genetic resources, here when we talk of plant genetic resources, we will concentrate on plant genetic resources for food and agriculture. And the management of these plant genetic resources, this broadly involves two basic components. One is conservation and then one is use. Here, actually, if we talk of conservation, basically, when we go for collection of plant genetic resources from the wild, or we go for their collection from different sources or already established sources, different gene banks, ultimately, they have to be evaluated in a local conditions. We have to go for evaluation of those traits which are not already available in our materials, and then those particular accessions and their genes identified, they have to be put to use for development of intermediate materials. So therefore, the pre-breeding efforts, they refer to all the activities which have been designed to identify desirable characteristics or the genes from non-adaptive materials. Then second thing is transfer these into intermediate set of materials. So therefore, an efficient plant breeder or an active plant breeder, he should or she should ultimately be able to manipulate the intermediate materials to develop new varieties. So here I would like to say that if there's any question, kindly note it down, and at the end of this presentation, we can take up few questions. So the point here is that the plant breeder ultimately, he will initially focus in the first step, in the pre-breeding step, to develop intermediate materials, or we also call it bridge material. And this material is later on to be deployed for development of new varieties by using this material into the main breeding pipeline. So pre-breeding is at the interface of conservation of plant genetic resources and plant breeding, and therefore it is a multidisciplinary endeavor, because here we require plant pathologists, entomologists, agronomy scientists, all sorts of scientists, even nutritionists, if you're targeting the nutrition traits, so that they can do an efficient evaluation of the plant genetic resources for us. Now, what is the need of distant hybridization and pre-breeding? Mostly, when we talk of the traits which are domestication related traits or yield traits, most of the times our objective is not to resort to the pre-breeding, because we want a time-bound solution there. We have most of the projects which are with the plant breeders or with us, they are generally time-bound. They have limited amounts of funds available to us, and then most of them are delivery oriented. But when we are saturated with all these resources with us, and we have plenty of resources, then we have to make a decision on that. Now it is the time to resort to distant hybridization and pre-breeding. And in this case, the objective is that we should focus on widening the genetic variability in the cultivated species. Then the modern day monoculture in many, particularly cereals, then if we want that our material, it has a broad base and there is not a resistance breakdown or tolerance breakdown, then monoculture has to be discouraged. The genetic erosion, which due to continuous cycle of breeding and inbreeding is occurring, that has to be tackled. And then we have to make available new genes in the cultivated species. If they are not available, then also we need to resort to distant hybridization in pre-breeding. Stress epidemics, generally they come due to, they ultimately culminate into the breakdown of resistances. Then of course, keeping in view the increasing food demands in the masses, and we are always in need of new species which we can domesticate and which we can put to food use, fodder use, and the feed use for animals. So neo-domestication is also one of the possibilities when we resort to the plant genetic resources, especially the wild species. And then in direct transfer of genes through breed species. So that's why the plant genetic resources, they for food and agriculture, they basically hold a great importance for all kinds of traits, including the different variations of traits which are still used by the farmers. Secondly, which are no longer needed by the farmers. And third thing is which might have previously been used by the farmers. And then the traits which have not been yet discovered or for new trait discovery. It is not necessary that whatever traits are either not available or they were available, but they are not in current need. So those species or those genes must be, they may be discarded. So we always have to keep safe with us all the genetic resources, all the genes which have been in use, which had been in use in the past and which will be of use in the future, so that as and when any need is there, or whenever any calamity stage is there, or whenever any exigency stage is there, then we can again mine these genes and put them into our genetic background, so that this can be more climate resilient, or the materials which we develop, they can challenge the need of the hour. So pre-breeding mainly aims at introgression and genetic base broadening. And when we compare breeding and pre-breeding, in a pre-breeding scheme, generally, it is in close collaboration with gene bank managers and breeders. So that's why in the Indian context, the importance of National Bureau of Plant Genetic Resources can never be undermined, and it will never be undermined, because ultimately, National Bureau of Plant Genetic Resources, they are the custodians of all the genetic materials as far as plant species are concerned in the country. And there has always to be a close association of breeders and the gene bank managers back at NBPGR. Then in the pre-breeding scheme, there's always a greater likelihood of more complex hybridization issues. Generally, linkage drags will be there. There are chances of reversal of the crosses to the parental forms. And then ultimately, there's a failure of the crosses. And then the end product of pre-breeding is a raw material for breeding. So here it is a very clear-cut definition, very clear-cut elaboration of what pre-breeding material, pre-breeding efforts result in. Ultimately, these pre-breeding materials, they result in the development of raw materials, which will be ultimately used in breeding. So this is the starting of another cycle. And breeding will ultimately result into a new variety. So generally, pre-breeding, actually, it requires a lot of patience, basically, because it is very slow, in fact, and time-consuming. There are very less chances of success as compared to the breeding efforts, and we generally say that it is almost 1/10 depending upon the species you are using. Then involvement of higher costs, because you have to resort to several kinds of new techniques, modern techniques like chromosomal manipulations, double haploidy breeding, tissue culture, then this kind of things, mentor pollen technique. This type of techniques we have to use, and it requires higher costs, and then manpower is also required in plenty. But the results are actually, there's a tremendous release of genetic variability, and on this genetic variability only we need to capitalize. However, this genetic variability is often very slow. The release of variability is often very slow, and it is not actually very powerful or very fast, so that we can immediately capitalize on that. Then selections are usually executed in later generations when we are going for pre-breeding, and the reason for this is that generally, in most of the cases, when we are going for distant hybridization and pre-breeding, the early generations, they always have, we are not certain in them, and there are always chances of reversal of the cross toward the parental line, especially the female line. And then special techniques are required most of the times, and then hybrid sterility and hybrid breakdown are the most common occurrences. So this is this was just the beginning of my presentation and a background. And actually, my purpose was to help you when we are going for pre-breeding and distant hybridization. The purpose was to help you in decision making. Our experience is, because we have been working with in close association with the NBPGR and the people working there in places like Kuldeep Tripathi, Padma Gore, and many other national and international partners, including our International Immunity Improvement Network, people from Punjab Agriculture University, TNAU. So many people are involved in pre-breeding. But what our experience is in general that whenever any new breeder or a young scientist, he joins the system, so out of enthusiasm, most of them, they want to capitalize on pre-breeding and they want to go for pre-breeding. But my dear friends, actually, when we make a decision on pre-breeding, basically we have to be very cautious on this. Cautious means this has to be actually very systematically done and very in a planned way, because if we immediately resort to pre-breeding, we need to see that whether our institute or a university can support our demands of high budget. Do we have an area which is designated for wild hybridization garden? Do we have laboratory facilities to carry out distant hybridization and then embryo rescue, chromosomal manipulation, mentor pollen technique, and all these things? Do we have adequate expertise as far as manpower is concerned who can carry out, for example, in vivo hormonal treatments and other techniques which are needed to support pre-breeding? So all these things require time, money, and energy. And if we do not have plenty of them, then it is most of the time it results in disasters. And most of the time it is a very heavy wastage of money, time, and energy. And ultimately we end up nowhere. So in that case, actually, we should take the help of the established pre-breeders for those people who are working with pre-breeding in the country. And we should, instead of requesting them wild accessions, we should request them to share their segregating generations, segregating materials, or the pre-breeding materials, which we can involve in the program. So in a nutshell, we can say that pre-breeding is not at all required if any one of the following is available. If we have commercially available, adapted and accepted varieties, there is no need of pre-breeding. Advanced selections well adapted to target environment. If we are already in F5, F6, F7 stage and we can develop a variety without resorting to pre-breeding, it's not required. And if we already have gene bank accessions that are well adapted to the target environment, even then we do not need to go for direct pre-breeding and distant hybridization. But however, we should resort to pre-breeding and distant hybridization when any one of the following is available. First is gene bank accessions are there which are not directly adapted to a target environment. Gene is there, trait is there, but it is not well adapted to target environment. Then if we have closely related wild species which are possible, then also we can go for pre-breeding, and wild species which are less closely related and more difficult to cross. So question is now with so many ifs and buts, who should take up pre-breeding and distant hybridization? So our experience says that advanced breeding laboratories with experience of raising wild accessions and exotic materials, they should have, and if they are having tissue culture facilities and controlled hybridization facilities, they have large experimental areas to grow the segregating generations, then they should go for pre-breeding. And it is not disheartening for others. Question is that if this is for the advanced laboratories only, then what other people will do? Other people in the country, they should depend on material sharing, raising segregating generations, executing selections, and backcrossing. Probably then they will be able to achieve increased success or higher success. And this is particularly important because if we take up the materials, because materials are very precious, we generally do not put any cost or any price to it, and that's why we do not understand their significance. Whatever material is available, whatever wild material is available, there are national repositories, there are national assets, and irrespective of not any cost being assigned to them, they are having tremendous cost. And if we are not expert and we are getting those materials, and if we are not able to flourish those materials in our wild gardens, this is a national wastage. So that's why those people who are not very much aware of distant hybridization and pre-breeding, initially either they should get trained in advanced laboratories or they should take the help of advanced laboratories to use this material. So now we come to the challenges of distant hybridization and pre-breeding. We generally face two different challenges at two different stages. One is vertical gene transfer and the horizontal gene transfer. So when we talk of vertical gene transfer, then almost always there are issues of crossability barriers, then chromosome pairing. Linkage drag is invariably in almost 80% of the crosses where we involve wild hybridization and distant hybridization. We are having, especially when we try to transfer genes from tertiary gene pool, then primary gene pool, then generally linkage drag is there, and it is then of course background effect and pleiotropic effects of alien genes. But when we go for use of tertiary gene pool, which we assign to horizontal gene transfer, generally regeneration protocols are the biggest challenges there, because the wild species from where we are transferring the genes, those species also do not have established regeneration protocols as far as tissue culture and embryo rescue is concerned. Many of the species, for example, in case of legumes, they generally show recalcitrancy and they are not amenable to they are not amenable to embryo rescue techniques or tissue culture techniques, and that's why success is not there. Then gene isolation from wild species, gene identification or gene mining from wild species, it's also very tedious task. Identification, it comes later on. In fact, what our experience says that growing wild species at the first instance itself is a challenge. And most of the breeders, they generally stumble for at least 2 to 3 years, 4 years before they can successfully grow wild accessions. Alien gene expression is sometimes it is suppressed, and generally most of the times, many times, some of the traits are there which are multiple gene controlled, which are controlled by minor genes and they do not express. And gene flow and associated risks. Yes, this is an increased area of concern of risk in recent times, that there might be some instances of gene flow and the associated risks which may be involved by use of wild species. But there are tremendous opportunities, and we should definitely resort to pre-breeding and distant hybridization. But in that case, we have to look at the opportunities available with us, and then what are the facilities available with us. Invariably, in most of the distant process, we require advanced in vitro techniques, especially the embryo rescue techniques. And sometimes there is a direct organogenesis, and sometimes there is an indirect organogenesis. And we need to be basically ready for both these kinds of techniques. In case of, for example, food legumes, because most of the species are recalcitrant, they used to be recalcitrant, although the recent protocols, they support the in vitro techniques, but still there are issues, and we generally most of the times have to go for indirect somatic embryogenesis or indirect organogenesis. I think somebody needs to mute his mic.
Kindly mute yourself, dear participant. Was just see who is there.
Then chromosome doubling in most of the cases, for example, in case of wheat, in case of triticale, in case of rye, generally when we are going for distant hybridization, there is an increased opportunity of getting haploids. And in that case, we should have an established protocol for chromosome doubling, and it is already there. Reciprocal crossing, breed species, and growth hormones. Basically, growth hormones we have experienced in many species where we go for pre-breeding and distant hybridization, that growth hormones, especially 2,4-D, and then gibberellic acid, this support this pre-breeding efforts. And there are different protocols for that. Sometimes we go for hormonal spray. Sometimes in case of cereals, we go for putting an injection of growth hormones at the base of the culm, so that the culm is filled with growth hormones, and ultimately it will lead to formation of embryo. Backcrossing. Then of course, yeah, cytology. One thing I should have told you in the beginning, that any person who is taking up pre-breeding and distant hybridization, he should have a support of very good cytologists, because ultimately whatever manipulations are occurring at the time of chromosomal cell divisions, then most of the times these cannot be detected at the morphological level, and we have to resort to cytology for studying their pairing pattern and all the hindrances which are occurring at the time of crossing over. Recombinant DNA technology is now very well being supported by the alien genes, and then cisgenesis and intragenesis, because there have been policy issues with recombinant DNA technology and transgenes. Then cisgenesis or intragenesis, where we go for resorting to recombinant DNA technology using the genes from within the species, which is widely acceptable. Then this technique can also be resorted when we are looking after the plant genetic resources, food and agriculture. Invariably, so nutshell, I can say that tissue culture and regeneration, they are basically a prerequisite for wide hybridization, and we should always aim at establishing very good tissue culture laboratory, or we can say that embryo rescue facilities, if we want to successfully go for distant hybridization and pre-breeding. Interspecific manipulation of chromosomes is one of the emerging areas when we go for distant hybridization. And this technique, distant hybridization, has also been successfully resorted for development of so many double haploid varieties in case of wheat, corn, and rice. And here, for example, in case of wheat, we go for crossing with maize, corn, and both of them are having the different chromosome number. And when we grow with corn, in fact, what happens is that there is an elimination of the haploid set of chromosomes of corn, and only the haploid set of wheat remains, and ultimately it will lead to development of haploids, wheat haploids, and these haploids can be subsequently subjected to this chromosome doubling using colchicine, and ultimately we get double haploids. And the most important advantage from this technique is that we can get a rapid generation advancement by instant fixing of the desirable this haplotype, and ultimately we get a diploid within a single generation, and we can save our 4 to 6 generations of sampling for getting a double haploid. So in this way, varieties have been developed, especially in India and China, and many of these varieties are quite popular in the seed chip. So here I just try to depict how, when we go for distant hybridization and pre-breeding, we can resort to DH breeding. This is an example of wheat. And here, first of all, if you see on the right of my pen, then left side of my this slide, you can see that a traditional cycle of crossing wheat variety A with wheat variety B, ultimately it will take 6 F1 generations, and then ultimately 4 years of testing, and then new variety. It will take around 10 years. But when we go for resorting to wheat into maize hybridization or wheat into Imperator cylindica hybridization, it will straight away reduce the time period from 10 years to 3 to 4 years. So there is a tremendous saving of time. So this is just again the depiction of shortening the breeding cycle. And here we have made a comparison of standard type of cultivar release using traditional methods and the double haploid breeding methods. And you can just see that if we take 12 to 13 years for successful release of variety in case of cereals using this double haploid technique through distant hybridization, or then we can release a variety within 8 to 9 years, saving a 4 to 5 years state. Now we come to the sources of genetic variation or gene pools for crop improvement. Although this is a very established concept of gene pools, so we are having different kinds of gene pools, and here I have to try to take one example each from cereals, then cotton, rye, means oilseed crops and pulses. So if you can see, we have just given few examples. For example, in case of common bean, Phaseolus vulgaris, which is in the gene pool 1, but it has other gene pools like Coccineus, then Costariceus, Polyanthus, and Acutifolius in different gene pools. Here, actually, our intention should be, whenever we are starting such a program, our intention should be to not go for secondary or tertiary gene pool. Always try to see the desirable genes in the nearest gene pool, for example, in the primary gene pool. And if we do not find any source of gene, a desirable gene in the primary gene pool, then we should resort to secondary or tertiary gene pool. So if we try to manipulate gene pool 1, gene pool 2, and gene pool 3, this is a generalized scheme where we can resort to utilization of availability of genetic variation in different gene pools. And here, if we see that if we go for utilization of gene pool 1, it is basically comparatively very easy, and we can go for rapid generation advancement using deploying different techniques like shuttle breeding, embryo rescue, hormonal manipulations, of course, day length modification. And then many times we are using indigenous methods also, like partial shading. You can see in the right side photograph, we are using simple net, green color net, which is available in the market for partial shading. And this has resulted in a very good success as far as crossing is concerned. And then off-season nurseries for rapid generation advancement we are using. And we are very easily deploying the gene pool 1, very regularly deploying gene pool 1 in development of improved cultivars. Whenever we are trying to use gene pool 2 and gene pool 3 for introgression, then there are several pre-zygotic and post-zygotic barriers. And these pre and post-zygotic barriers, they pose tremendous hindrances, but ultimately we have to support this type of hybridization by different techniques like delayed pollination. We have found that we go for emasculation or removal of anthers, and if we delay the pollination, then the chances of success increase. Partial shading, I have already told you. Mental pollen is a very established technique where we use a mixture of pollen, and it is an age-old practice, maybe being used in the last 50, 60 years. And what we do is we make a varietal or species mixture of the pollens, and then we go for using this pollen for hybridization. And generally, this technique is a hit and try technique, but it gives a lot of success also. Sequential embryo rescue, I already told you that it is a must basically, and we always have to most of the time resort to embryo rescue for the recovery of our interspecific hybrids. And hormonal manipulations and utilization of gibberellic acid and 2,4-D for in vitro or in vivo hormonal manipulations has been found to be tremendously successful. One more important thing is that pre-breeding and distant hybridization, they not only result in development of large, means a huge segregating population or in the breed species. These have certain other advantages also, like we can also use these techniques for development of advanced backcrossed QTLs, then introgressal lines, chromosome segment substitution lines, and chromosome substitution strain. So most of these kinds of populations, they are basically quite useful for different kinds of molecular studies, as well as basic studies. And these populations can be very effectively deployed for identification of QTLs controlling complex traits, then the genes which are which are controlling simple traits, and likewise, and later on indirectly, this information can be used for development of different kinds of populations and building materials. This is just an example of broadening the genetic base of pigeon pea through distant hybridization. So this is a generalized scheme that we have n number of genetic resources available with us. So many wild accessions, cultivated accessions, exotic lines, land races, so many things. In fact, we have to pass all this material through a selection sieve or evaluation sieve, and only those materials which fit the criteria of being easily crossable, then of having the gene of interest in a tremendous or very robust intensity, and then expressible. So these criteria, if it fails, and then we have supporting techniques available with us, we should identify a species which can be deployed, that can then be put to use for pre-breeding efforts. And this pre-breeding efforts, typically it will take 5 to 8 years because it requires a lot of patience while maintaining. Sometimes we have also experienced, our first-hand experience is that we made so many crosses, and after the end of 5 or 6 years, we were nowhere. All the crosses, either they were lost, they were different kinds of problems like hybrid sterility, hybrids are not regenerating. Sometimes we lost even the segregating generations. When we isolated the plants, they reverted back to their parental lines. Then hybrid death, then leaf necrosis, and these kinds of problems will keep on occurring. And ultimately, a breeder may get easily frustrated after a cycle of 5 to 7 years, and he will get nothing. But sometimes, if we are lucky, then even in the second, third generation itself, we may be able to find certain plants which can ultimately be released into varieties. And I will cite you some examples. For example, in case of mung bean, some of the most popular varieties have been developed through distant hybridization, deploying Vigna mungo and Vigna radiata gene pools, and successful crosses were made. Now we have Vigna sylvestris also in the products in the pipeline. We have Vigna umbrata in the pipeline. We have Vigna angulata products in the pipeline. So it depends upon the species you are using. And the second thing is compatibility amongst the different species. Then cytogenetic approach for identification of alien chromosomes and segment. We have done our job already. We have made successful crosses. We are at the advanced stage. But how to make us sure that how we are going to identify the alien chromosomes and segments? Of late, although this particular slide is a bit old, and this I deliberately show because this is one of the first slides of deployment of fluorescent in situ hybridization techniques, GISH and FISH techniques on the metaphase spreads of critical and wheat, where besides the molecular techniques using the cytogenetic techniques, cytological techniques, the people, they were able to identify and detect the alien segments. For example, here you can see in the top slide and in the bottom slide, which have been marked by the arrows, the alien segments which were discovered in the products which were developed through distant hybridization. And this technique has of late developed very much, especially in some places like University of Leicester and Cambridge, and they are deploying these techniques for successful detection of alien chromosomes. So this was the basic information about pre-breeding distant hybridization. What should we do? What should we not do? when we should resort to pre-breeding and distant hybridization, what materials we should use, how we should proceed ahead, and what we should do when we are not successful. Now I will be coming to the second part, which is more of practical part and our experience, first-hand experiences I would like to share with you, that where we have been successful, because I am primarily working right now in pulses. So I have ample examples of pulses to cite that where how we have been successful using pre-breeding and distant hybridization. I need not give much introduction to you about pulses, as you know that these are climate smart, nutrition smart crops. High protein is there, high nutrition is there, their proteins are of high quality, and they are basically the cheapest source of proteins. And they also add to soil by way of increasing the soil means nitrogen, and then the soil carbon also, because they aid in higher soil carbon sequestration, and this also because their root system is so that it reduces the risk of soil erosion and depletion. These are just two slides for passing slide to emphasize that why we need pre-breeding efforts in pulses. Actually, when we see pulses, because about 1 decade back, we were producing only 14 to 15 million tons of pulses, and we were short by 8 to 9 million tons of pulses at that time. And we were importing pulses to the tune of 18,000 crores of Indian rupees at that time. So when we made a policy papers about increasing self-sufficiency in pulses, so one of the major things which came out was there were actually 3-pronged strategies. One was vertical expansion, second was horizontal expansion, and third was reducing the crop duration and reducing the post-harvest losses. When we talked of this vertical expansion, the major initiative to be taken there was that we should increase the genetic potential of the yield potential of the crops. So if we increase the genetic potential by 30%, and even if then there is a gap of realized yield and the potential yield, realizing is in the farmer's field and the potential yield, but if we increase the potential by 30%, so at least 20% production will increase. How to achieve this 20% increase in the production? So this could be achieved actually by deploying certain genes, or at that time we said that there has to be a kind of disruptive breeding. There has to be very aggressive breeding, basically using the wild genetic resources, exotic resources, land races, whatever available with us, so that we can pull up n number of genes in our material, so that the material has a lot of genetic potential. And even if there is a gap, the gap continues, the present gap, realized yield and the potential yield, the gap, it continues, even then the yields increase by 20%. Keeping this in view, the need of capitalizing on wild genetic resources was also realized, and it was targeted that if we start it now, in next 10 to 15 years, we will be at a certain page to utilize the wild genetic resources. So these were extensively taken up in means pulses, in different kinds of pulses, and ultimately we are now at a certain stage that we are having in most of the pulses, including chickpea, including Vigna crops, then in pigeon pea, we are having products now, we are having materials now which are either at the intermediate stage, or these have already been introduced into our breeding material, and we are now releasing advanced materials and varieties for commercial cultivation. If we see Vigna crops, the Vigna crops including mung bean, cowpea, urad bean, and several minor pulses, there are actually these are some of the pulses which are grown in wide tracts of India. And because these are indigenous to India and their origin, center of origin and center of diversity, they are located in the Indian subcontinent only. So these are indigenous ones, and we have plenty of genetic resources, wide genetic resources available with us for improvement of these crops. Their major problems are in diseases: yellow mosaic disease, leaf twinkle, cercospora leaf spot, powdery mildew. There were problems of pre-harvest sprouting. There was a problem of photothermosensitivity. Then there were the problems of insect pests like thrips, whitefly, defoliators. So these kind of problems were there. And about 10 years before, or a decade before, most of the materials which were having, they were shuttering type of materials. The plant habit was trailing. The plant was trailing, and there was no determinacy. We had to go for multiple pickings, and there were so many issues. If you see current day scenario, in last 10 years, whatever varieties we have developed in Vigna crops, almost all of them are totally free from yellow mosaic disease. The plant type has been changed. most of the varieties have become determinate, they mature together, and there is no need of multiple picking, and the yield potential has increased. So if we talk of Vigna species, it is Vigna genus, it is having more than 200 species, and amongst these 16 are cultivated for human consumption. And many more are there which are used for fodder and animal feed. So as far as why I am focusing on Vigna is because these are indigenous crops, and the crops which are actually in India or the Indian subcontinent, in the Asian subcontinent, in the Asia continent, we call them Asiatic Vigna. And basically these are in subgenus Ceratopis with 21 species, and which generally the morphological differentiation is quite limited. So we say that it's a homogeneous group, and these actually, because these are indigenous and we have a lot of plenty of materials, these offer tremendous scope of trait improvement through wide hybridization. So we collected a lot of species together with MBPGR. We conducted many exploration trips in the Western Ghats, in Himalayan regions, in northeast part of India, along with NEPGR Regional Station Trichu, NEPGR New Delhi, and other stations. And you can see here that all of these species were very systematically evaluated for different traits, all traits such as bouquets, powdery mildew, low trypsin inhibitor activity, heat tolerance, salinity stress, salt stress, so many things. These were evaluated, and we were able to document a number of species which have been reported possessing these traits in intense form. So this is just a pictorial type of thing to just show you that how we did these together along with National Bureau of Plant Genetic Resources. And we conducted many such trips and collected a large number of wild accessions or the accession in the primary, secondary, and tertiary gene pools. And you can see on the right pan that we took help of local farmers also, even the forest people also. And on the left side, you can see the variability available. So this is just a photograph of wild hybridization garden, which is established at IIPR Kanpur, which contains more than 200 accessions of 21 different species of Vigna. A similar garden is now established at our regional stations in Dharwad and Bhopal. Likewise, National Bureau of Plant Genetic Resources also has its own wild hybridization garden, and they are having all these accessions here. Likewise, you can see here that such accessions are collected from the roadsides. Here you can see that we also collected wild accessions, wild relatives of Cagenus like Rincosia, then Cagenus lineatus, then Cagenus scaraboides, Rincosia hainii, like this. And ultimately, we have been able to establish successfully the wide hybridization gardens at IIPR. Then a beautiful garden is available at National Bureau of Plant Genetic Resources, many of our regional stations, and now we have a very rich repository of wild accessions available in most of the pulses. So at the bottom, you can see that you can see a very nice picture of the wide hybridization garden of Vigna at IIPR Kanpur. So yes, when I was discussing about raising the wild accession, and I was requesting that everybody should not resort to distant hybridization and tree breeding, the basic reason behind that is now you understand that how intricate it is, that unless we are using elaborate techniques and properly subjecting the materials for scarification, raising of the material, and then means its proper care, then we will lose the material, and basically this will be a national loss. For example, invariably in most of the accessions, seed scarification is required, which is done manually, and each seed has to be manually scarified so as to break its seed coat so that water imbibition is proper. You can see in the right side photographs that how we have gone for scarification of the seed. And of course, this has led to increased germination. Here I have compared also that how much improvement in seed germination was recorded in different species. Then the second step always is, once we are able to successfully establish the species and we are able to take good care of them, they have to be subjected to morphological and molecular characterization. And morphological as well as molecular characterization are most important, because characterization at the morphological level may sometimes be misleading, as many of the genes they either do not express, or most of, because you know that Vigna species or pulses in general, they have a very high genotype into environment interaction influence, and most of the times environment has a very, very huge impact on the expression of these. Yeah, so we did an extensive evaluation of these Vigna species at the morphological level, and it's just to show you the difference in different extent. For example, you can see in the top pen as well as the bottom pen, hypocotyle color and growth habit. You will see that there's a tremendous difference in different accessions for these two traits. In the bottom, in this slide also, we have done evaluation for corolla color, resting position. Then you can see the variability in pod, shape, size, color. Then this hairiness. So a lot of differentiation is there, and their smoothness, curve, seed size, seed shape. So you will find all sorts of characters, all sorts of traits within these species. So you can see here that the difference, genetic difference for pod color maturity and pod constriction. Then of course, yes, seed color, seed shape, size, luster, then the shape of the ripened pod. So this kind of morphological characterization was done continuously for 3 years. And I'm happy to say that the NBPGR scientists, especially Dr. Padma Bole, was involved in this kind of Herculean task with me, our team. And we conducted very extensive experimentation of morphological evaluation of these species at IIPR Kanpur as well as NBPGR and our regional stations. And then we were able to bring up a very comprehensive and extensive data, which has been published in very good journals like Frontiers in Plant Science, PLOS One, DMC Plant Biology, and several journals. We have reported all our findings, and these have been cited widely. Followed by this, we went for genetic diversity, and actually this is just a study, but mainly speaking, we went for molecular evaluation also. And till now, we have resorted to only the SSR markers because as far as Vigna species are concerned, we have a dearth of the SNP other platforms till date. So we focus on SSR markers, and we went for an extensive molecular characterization of all these species. And on the basis of that, then we went for further studies like population genetic structure analysis, genetic diversity, and marker trait associations. For example, here you can see that we tried to associate different, using these species, we went for association studies, and different kinds of traits, markers were associated with them. And these markers now we can deploy for our marker assisted breeding program, as in case of chickpea we are doing, and we have released varieties in chickpea. So this is with another set. For example, for yellow mosaic India virus, we were able to associate 3 markers on the basis of molecular characterization. Then this is just to show you that different kinds of marker studies we carried out. Then the second step from our morphological evaluation was to identify trait-specific donors. And trait-specific donors means because there were some traits for which we were not finding a robust kind of resistance in our cultivated species. And bruchid was one of them. You know that bruchid is one of the most notorious pests, storage pests in pulses. And sometimes the damages are 100% because it will bore a hole into the seed and it will make it hollow, and it will render the seed unfit for human consumption. We were not finding many species in the cultivated, and then we had to resort to the wild ones. And I'm happy to share with you that in the last about 8 months or 1 year, we have been able to register with NBPGR 9 different species accessions, in fact, for resistance to bruchids against the species Calosobruchus maculatus and Calosobruchus chilensis. We are struggling with Calosobruchus analis, and we hope that on the basis of our studies till date, we'll be able to identify the resistant species against Calosobruchus analis very soon. Then we also identified accessions of Vigna umbellata and Vigna glabrescens, which were photo and thermoperiod insensitive. Likewise, for Vigna radiata, for yellow seed coat color, we call it golden moon. We were also able to identify and characterize Vigna accessions for photothermal insensitivity. Actually, Vigna species are highly influenced by the environment, as I told you earlier. But we found 2 accessions, one each in Vigna umbellata and one in glabrescens, which can flower and pod between the temperature range of 4.4 degrees centigrade to 44 degrees centigrade. So it is just marvelous because especially during when the temperature goes less than 20 degrees centigrade, it's very difficult to have the Vigna flourishing. So these accessions now we are using very widely in developing photo and thermo insensitive varieties. Then besides this, again, we went for yellow mosaic disease. It is one of the most important diseases and in Vigna. And this disease may also lead to tremendous losses, sometimes 100% loss of the crop, besides other diseases. So we have been able to identify several species which are actually highly resistant or moderately resistant. I was talking about broochids. So this work was extensively done by one of our scientists, Dr. Revan Siddha, who is an expert into brookid screening, and he has been able to report many species which are actually resistant to brookid. Then this is just an example from other crops also. As I was telling you that distant hybridization and pre-breeding, it may lead to development of, it may lead to other studies also simultaneously. For example, I have cited here the advanced backcross QTL analysis for wild QTL integration in legumes. And once these QTLs have been identified, they can be deployed for improvement of the cultivated yields. One very important thing here you can witness from the photographs is you can see that how intense particular traits, how intense we can get it from introgression of wild accessions. For example, here you can see the number of pods in some particular segregants or some particular derivatives which we obtained from wild and cultivated crossing, especially in case of mung bean and urad bean. And even we have got some particular plants where we could witness more than 500 to 600 pods per plant, and it was tremendous. So then we used all these accessions, as I told you earlier also, and now we are at the advanced stage. Like we are having crosses at different stages like F6, F4, F5, and we are having now a lot of materials which have been developed using wild crosses, and I'm quite hopeful that these materials will either be put to the main breeding pipeline within next 1 or 2 years, or a few of them where we are finding very good adaptability will be put into the evaluation pipeline. So these are just a few slides showing how good segregants we are obtaining using the wild accessions. So we are also having some kinds of mapping populations also, then varieties. For example, there's a I would like to cite an example of a recent variety developed using mung bean and urad bean crop. This is Vasudha or IPM 312-20. So this variety has been specifically released for summer season and suitable for Uttar Pradesh. And this variety is a result of interspecific hybridization. Then other varieties which have been developed successfully in case of Vigna are, for example, Kanth Mung 4, then HUM1, although these are old varieties that are now out of seed chain, but these are the results of distant hybridization. Meha, developed by IIPR Kanpur, has been one of the most popular varieties of mung bean for Kharif season, and it was developed using the cross of Vigna radiata and an amphiploid of Vigna radiata and Vigna mungo. Likewise, other varieties. And then of late, you will see that in case of black gram, there are several varieties, 3 to 4 of them, which have been developed using Vigna sylvestris as the donor. So my point here is that not only urad bean, but we have been able to utilize the gene pool of Vigna sylvestris also. We have several materials which we have developed using Vigna ambulata, in Vigna anguiculata, and several other species. So here is another variety which has been developed by Punjab Agriculture University Ludhiana, which is SML 827. And you can see that it is an introgression product by using Vigna radiata ML881 and Vigna ambulata. So this is also one of the popular variety which has been developed only recently using the interspecific hybridization. Vasudha, I have already told you. So this was about some successful examples. I specifically cited the examples of Vigna crops because in Vigna crops we have our first-hand experience of deploying distant hybridization and breeding. Now, what are our future thrusts? Probably this is, I think, last but one slide. Yeah. So future thrust will be the first thing is for trait discovery. Actually, as far as most of the yield related traits, the nutrition traits, and the domestication related traits, we have ample genes available for them in most of the crops, not only Vigna crops, but also other crops like cereals, oil seeds. But it has to be done for specific new trait discovery, and we have to find some other traits which are actually not available in intense form in the cultivated species. Then the missing traits for the future traits. Here we have to define future traits. For example, one can be supernodulation impulses. We can increase the number of nodules. Or, for example, in case of Rajmarsh, there is no nodulation. We can introduce nodulation in Rajmarsh. So this type of traits we have to focus. And the crop missing traits, when we talk of ideal plant type, there are certain traits which we need to focus upon to improve. And what we feel is that the plant type which has been defined 20 years back or 30 years back, it also needs to be changed as per the need of the time, because now we have seen a change in the climate. We have seen a change in the need of the plant type, and that's why we have to tailor plant types. So we have to define new plant types which are suiting to the current needs. For example, in mung bean, the new traits means the traits which we can target using the Y species can be root tolerance, cold tolerance, podding from the base or bodial podding, then mechanical harvesting, of course, biofortification, high amylose content. Likewise, in case of urad bean, it can be early growth vigor, long pods, more seeds, tolerance to high pH, high methionine, and tolerance to high temperature. So this is my last slide. I think I am catching the time. I would like to thank sincerely our collaborators, especially our regional stations, PA Ludhiana, IRI New Delhi, NBPGR New Delhi, Badnapur, and TNE Wamban, ACI Taiwan, and Hyderabad, then World Watch Hyderabad, ACR Australia. Actually, it used to be ACR Australia. I am sincerely thankful to our funders, including ICR, the NICRA project, which has supported us for taking mung bean research in the country to next sites, DBT Government of India, New Delhi, ACIR Australia, Lucknow, and we are a huge team working back at Kanpur. I'm extremely grateful.
Thank you very much, sir, for making us aware about the significant contribution made in the Vigna under your leadership, and you have really reshaped the Vigna breeding program. So shall we take, sir, some questions?
Yeah, definitely.
Just I'm taking from chat box. Now, Dr. Vishnu Gite from Parbhani, he asked that he is facing the problem of pot setting in interspecific crosses of rice bean and mung bean. So he's asking that which growth hormone at what concentration should be used for pot setting and this thing. Whether you are taking, we can take the colchicine treatment and this thing. He's asking like this.
Yeah, I think colchicine treatment will not help you at this stage, because colchicine has a different kind of mode of action, and it is mostly used for chromosome doubling. So it will not be useful. But if you go for spraying of gibberellic acid at the rate of 1 ppm, 24 hours after pollination, this will definitely help you. So what you have to do is that once you pollinate the flower bud, you just because we always go for tagging of the flower bud, and we have to make a 1 ppm solution of gibberellic acid, and if we spray it continuously for 3 days, 24 hours after every 24 hours, it will be helpful. Second thing is partial shading is also helping in such kind of crosses. If you go for partial shading, and the moisture should be plenty. It should not be very high, but it should be sufficient so that there is no heat stress.
Question from Dr. Sheetal Patel. In mung bean, generally we have hard seeds. What is the reason for these hard seeds?
Yeah, hard seeds generally, hard seeds we get during the summer harvested crops. And the main reason behind this is forced drying. Basically, what happens is at the time of maturity, especially when it matures in May end, or when the temperatures are more than 45 degrees centigrade, what happens is the seed, it all of a sudden dries due to the heat waves. And this kind of seeds, they develop into hard seeds. And generally, we find about 2 to 3% hard seeds in the summer crops, which are harvested during the months of May or June, till June 1st.
And also Dr. Suma Mowgli from, I think, Dharwad. It is a transient. It will be resolved. It's most of the genomes, right? And next question is by
primary, secondary, tertiary.
Dr. Thangovendan, difference between primary and secondary tertiary. It's a very obvious, I think.
It's a very basic question. Actually, there are definitions available on Google and all graduate books. So basically, the primary, secondary, and tertiary gene pool, it has been defined on the basis of crossability status. And then different criteria also followed for this. And I think you can just, for a definition point of view, you can just search Google.
And Dr. Sunil is asking, what is the minimum population size and marker density for precise mapping of genes in association mapping?
Actually, as far as association mapping is concerned, the population size and marker density depends upon the species you are dealing with and its chromosome number. Normally, we say that, of course, population, what type of population you are using? You are using F2 mapping population, you are using backcross mapping population. So it is different for different. But normally we say that for a self-pollinated crops, and generally where the chromosome numbers, they vary from 20 to 30, the ideal population size should be more than 500. So 400 to 500 should be the ideal population size. But in most of the cases, generally the marker, and then it also depends upon the representation of your samples. If you are having a wider genetic variability within your samples, then then the population size can be a bit small. And if it is about 350 to 400, it will also be equally.
Okay. And Dr. Malvariji Devarajan from TNU, she's asking like how screening for brucids is carried out.
Yeah, actually there is an established protocol for it, and I will give you some reference or papers which we recently published. As far as screening is concerned, normally we depend upon free choice test, no choice test, and then confirmatory no choice test. This is basically a 3-step procedure. For free choice test, first we go for free choice test, and where we allow all the brucids to feed freely on the particular a mixture of varieties or a mixture of species to feed upon. And there is a standard protocol for it. We release males and females of brucids into a vase, and then we put the seeds here. Out of that, which ones are resistant, we identify them. Then we go for no choice test. We do not give any much choice to them. We go for force feeding. We allow them to force feed on the materials which we have provided. And once we are able to identify particular species on the basis of both the informations, then we go for confirmatory no choice test. So this is a 3-step procedure, and it is established.
And Dr. Anupama Singh is curious to know any accession for PHS tolerance or resistance in mung bean and urad bean.
It is difficult to say right now. We have started the work on pre-harvest sprouting tolerance. Actually, most of the materials, they generally resist pre-harvest sprouting till for around 20 to 24 hours of showers. But beyond 20 to 24 hours of showers, the mung bean, they generally start sprouting. And as of now, we have started work, and probably I cannot say with confidence that we have any accession available as of now. And we are working on that.
And one more question from Dr. Mamta Singh from NBPGR. Is it possible to get rare combinations in pre-breeding segregating material, which were not originally available in parents?
Yes, definitely we can get it. That's why we are doing pre-breeding. You must have heard about transgressive segregants. So transgressive segregants, it's always a chance to get transgressive segregants also. And sometimes, for example, generally for maturity duration and such other traits, we very regularly get transgressive segregants, and we can definitely get them. The only thing is that how keen an observer is in the field while we are making selections.
One more question, because I think your talk is very relevant. The participants are finding relevant, and also they are curious, and they have the Dr. Chandana Behra. She's asking like, I have some wild species of mung bean, but I don't know exactly the species name. So definitely, if you have, I can answer it. If you have wild species, either you can share with us also. We'll be able to find out. And Dr. Anupama Singh is asking, sir, how we can do screening for PHS if it is not available in field?
Yes. Actually, PHS ke liye, there are some literature is available. In fact, pre-harvest sprouting occurs when there is a drizzling for around more than 18 hours continuously, and the relative humidity in the atmosphere is more than 90%. So that is the most ideal condition of PHS. So when we are going for pre-harvest sprouting, what we do is we generally create such kind of conditions in the lab. And as of now, we are using the Petri plates for that, moistening the paper, we have filter paper, we are putting on the base, and then we put in the incubator, maintaining the maintaining the moisture content more than 90%. And then artificially we are doing like this. Actually, the thing is that we have to maintain the moisture content in the seed as well as in the atmosphere, so that the ideal condition pre-harvest sprouting are there.
So I think, sir, question is over, and you have nicely explained all the queries of the participants. So with this, I would like to thank you, sir, on behalf of my own and organizing committee. You have, I know you have been, quite busy since last 2-3 months, but you have accepted, and you have landed in Kanpur, and you have agreed for us. So thank you, sir, for once again. I hope all trainings have benefited. I will share your contact details to them. if they are having any query and this thing, to definitely they will be benefited with your guidance and mentoring. And you are the person which are not just many of the person. Of course, I am following your footsteps, and you are really, source of motivation, not just for many of person, and of course for me also. Thank you, sir. Thank you with this.
Thank you for your nice words. Thank you, everybody. If you still have doubts, you can contact me personally on my WhatsApp or email, whatsoever. Thank you very much.
Thank you. Thank you, sir. Now I'd like to Dr. Subhas for further.
Yes, sir. Yes, doctor. We have further we have lecture by Dr. Sanjeev Saxena, sir, on intellectual property right in PGR management. Sir may join within 4-5 minutes, then we'll start. Good afternoon, sir.
Good afternoon. Good afternoon.
And welcome you, sir.
Yeah.
Dear participants, our today's speaker, Dr. Sanjeev Saxena, sir, has joined with us. Please put on your videos.
ठीक है, एक ही साल में बना, ठीक है।
Friends, we are with our former Assistant Director General, sir, Intellectual Property Right, Property and Technology Management.
अभी शेयर नहीं किया मैंने।
I formally introduce to you all. Sir has a very long career of more than 29 years in the ICR and other prestigious institutes of India. He has been involved in different dimensions of biodiversity, genetic resources, intellectual property, and technology management. This include analysis, development, and implementation of policies, advocacy, and awareness generation. His involvement includes other related dimensions with plant variety protection, seed sector issues, farmers' rights, biotechnology, environmental concerns, and traditional knowledge. Sir has been closely associated with several reforms related initiatives in ICR. Besides this, he is being instrumental in the formation of several mega projects on crops and seeds, research and development. Sir, I again welcome you, sir. And sir gave a lecture on intellectual property rights in PGR management. Sir, I request you, sir, please enlighten us with your lecture, sir. Sir, please share your slides.
Thank you. Can you listen to me?
Yes, sir.
Okay. It's always been a pleasure to be with NBPGR, where I worked for more than 14 years.
Yes, sir.
Thanks to Dr. G.P. Singh, Dr. Subhash for the nice introduction. Well, the topic today is PGR management and IPR. I'll just try to share slides. Are you able to see the slides now?
Yes, sir. Yes, sir.
Okay. So when we talk of IP laws and agriculture research, up to 1985, so it was an era of free exchange of plant genetic resources and technologies. And we had to see no obligations, and all the world benefited with that free exchange. People could go anywhere, collect the germplasm, use it, and then generally make products and valuable things out of it. From 86 onwards, there were many developments which took place. And largely they were Convention on Biological Diversity and the WTO, World Trade Organization, which was formed in the TRIPS negotiation for intellectual property. Now, the nations were obliged to have, during this period, national laws which were compatible with the obligations of these treaties. Among others, for us, what was more important was the issues regarding access to genetic resources, benefit sharing with the farmers and the community, the rights of the farmers, and also traditional knowledge and similar issues. They were very well deliberated, but then from 96 onwards, as these international developments firmed up, we also had to have our own national laws, and PPBFR Act, that is the Protection of Plant Varieties and Farmers' Rights Act, and then the Biological Diversity Act, which regulated excess land, actually, were enacted, and also the patent laws and other IPR laws were amended or enacted, which were to be compatible with the minimum standards as per the TRIPS agreement. Just a view how it all has made things more complicated for us. International undertaking, that is what I was talking about. That we considered it as a common heritage of mankind, all the genetic resources. and we all worked under the international undertaking on plant genetic resources. However, 93, as we said, the Convention on Biological Diversity was in place, and then it said that nations have a sovereign right on the genetic resources with them, and conservation, sustainable utilization, and equitable benefit sharing were the main pillars of the Convention of Biological Diversity. But being sovereign right of the nations brought in the concept of ownership of the genetic resources occurring in a particular nation. And the Convention on Biological Diversity comprised of all bioresources and not just the plant genetic resources what we are today interested in. So for this conservation, sovereign rights, and regulated access to all bioresources, we have the Biological Diversity Act, which was enacted in 2002 and rules 2004. So this act came into being. Now, there were many genetic resources which were already conserved or already available with the countries outside the domain of the country where they were collected from or the country of origin. So they were in the form of ex situ collections. So there was a lot of discussion about the gene banks who were having the PGR and ex situ collections and how there would be now sovereign rights of the nation would be expressed. So it was decided to have the International Treaty on Plant Genetic Resources, which came out in 2004. And it said about the facilitated access and a multilateral system of use on mutually agreed terms and based on standard material transfer agreements. But then if you see, an independent development was taking place at that time. In 1994, the WTO and TRIPS came in, and it made mandatory requirement for protection of plant varieties and also made microorganisms as patentable. Protection means intellectual protection for plant varieties. And therefore, we also had the act in the national system, the Protection of Plant Varieties and Farmers' Rights Act in 2001 and notified in 2005, which has IPR protection for the varieties. There were countries which were giving a patent type of a protection to the plant resources or genetic resources. And also there was an existing system of UPOV at that time. Now, although WTO was a totally independent occurrence as compared to Convention on Biological Diversity, but the problem came that products and access were through the International Treaty on Plant Genetic Resources and Biological Diversity Act, but they were amenable to patenting or protection of IPR. Therefore, there was a concern where both these things were intersecting. More so because our Biological Diversity Act, as compared to the other, was also including benefit sharing for the community as per the CPD, right? Also, our BPVFR Act also talks about farmers' rights. So that is the complexity whole, which I think when we talk of genetic resources and IPR, that as a student of genetic resources and IPR, we should all be able to understand the intricacies. Now, before we go deeper into it, let us define an IPR. IPR is something which comes out of your intellect. It is intangible. And when it becomes tangible, it becomes a property. It does not diminish with use. It becomes outdated. Like if anything you create from your intellect, it does not diminish with use, right? It may become outdated or version may change. And then the third component is rights. Rights are something which the state or the government can only give. So the state gives, it's a legal domain, and it's a sort of a monopoly right. So the basic features of IPR, all type of IPRs is that they are statutory rights. Means there is a statute or a notification, gazette notification through which this right is given. It is territorial in nature. Obviously, every country has to have a law, and that law will be applicable only in their territory. Therefore, it is territorial in nature. There is nothing like a global patent or global IPR on anything. You have to go to every country and take the IPR there. Now, what does it do? It provides a sort of monopoly to the right holders for commercial exploitation. What monopoly? That it enables holders to exclude others from authorized use. So basically, you can say it's a negative right. If I have an IPR, I have the right that nobody else can use that intellectual property without authorization from me or the holder of the IPR. And it is always given for a limited period. There is, because it's a monopoly right and given for people to have commercial benefit out of their invention or the new intellectual property which they have created. So therefore, it is always for a limited period. Now, as I said, the intellectual property right. So the intellect can be manifested in many ways. You know? So it can be in the form of an innovative product or a process. It can be a cultural, artistic, or literary work. That's also, if I write a poem or have a movie made, it is also a sort of an intellectual input which I am giving there. It can be a creative design, and it can be a distinctive sign or a denomination of goods that attributable to a geographical origin, or a confidential business information, or even new plant varieties. So what I'm trying to say is that there is no one shoe fits all IPR. For every domain of your intellectual creation, there is this appropriate IPR. Like for products and processes or inventions, it is patents. For cultural artistic works, it is copyrights. For designs, it is industrial designs. For distinctive signs, trademark, you all are aware of different trademarks available, and denominations of goods attributable to geographical region. This directly is not related to plant genetic resources, but yes, agriculture products are amenable for geographical indication. Means if a product is having its attribute due to the origin in that particular geographical region, then it's a collective mark, different from trademark. Geographical indication rights is, number one, it is a collective mark. So the producers or the manufacturers of that region, of that particular geographical indication, have a collective right. How is it different than the other rights? The other rights, in fact, have the philosophy that somebody creates a new intellectual property, discloses it, and puts it in public domain. So he should, or she should, get some sort of a intellectual benefit out of it or a commercial benefit out of it. Geographical indications rights is slightly different in connotation. It is more for the consumer. Suppose I want to have Banarsi sari, right? So when I go to the market, there will be so many people having the label Banarsi sari. But how do I know that the Banarsi sari or the Darjeeling tea which I am taking is really from Banaras, made by those artisans, or the tea collected from Darjeeling? So the people who are having the location specific people are identified who are given the geographical indication logo of that particular product. So therefore, if I have a tea which is a Darjeeling tea and it has a location of GI over it, then I know that yes, this is created by an authorized person who is there in Darjeeling and collecting this tea. Similarly, for the Banarsi sari. So it is more for the benefit of the consumer, but no doubt about it. Once it has got a market, I want only Darjeeling tea, certainly it gets a premium to the people who are producing it. So that way it is slightly different than the other IPRs. And confidential business information is trade secrets. You all know that if this Coca-Cola is an example given so often, nobody knows the formula, and it has been protected as a trade secret. And Plant Varieties Act, as we know that patents and plant varieties among all these, apart from geographical indication with a different connotation, are the acts which I think we will have to go through slightly in detail to understand how the genetic resources and IPRs are related. Let's go for patentability. How can you patent something? You have to first see if it is novel, that must be new. Is it inventive? New means there should be no prior art existing on the product or process which you have invented. Inventive means that the person trained in the art should see it, that there is a technical advancement as compared to the existing knowledge. And is it industrially applicable? That is, must have utility and should be vendable. Therefore, if these three criteria are met, then you can get a patent. And period of protection is 20 years. As I said, they all have a limited period of protection. So it is 20 years. Now, the Act of Patent also says that there are things which cannot be patented. And here we are more concerned because we are dealing with plant genetic resources. And I'm restricting to that area. One thing it clearly says, method of agriculture or horticulture cannot be patented. Second, it says that plants and animals in whole or any part thereof other than microorganisms, but including seeds, varieties, species, essentially biological process for production or propagation of plants and animals are not inventions. So they cannot be patented under the Indian Patent Act. But as you see here, microorganisms can be patented. But here also you should be very clear that microorganisms as they occur in nature are not patentable. They are patentable if they fulfill the criteria of patentability. Means it should be novel, the process of the product which you are generating out of the microorganism should be novel, there should be an inventive step, and it should be industrial applicable. But yes, per se, microorganisms cannot be patented. Section 3P says that invention which in effect is traditional knowledge or which is an aggregation or duplication of known properties of traditionally known component or components is non-patentable. What means to say that? You must have all heard about the Haldi patent and all those things which happened at that time when we were beginning and entering into the IPR era. So it clearly says that any traditional knowledge for which there is an aggregation or duplication of known properties, so you can't protect Haldi for wound purposes or similarly other things which are already mentioned in our traditional literature. So if you want, you can go through and search on the website about TKDL, Traditional Knowledge Digital Library, which was an initiative taken at that time when the Haldi patent started and we were all into the game of it. The CSI started it, that they converted all the traditional knowledge in Ayush and Ayurveda into patent searchable form. And therefore they were made available to the patent offices. How do the novelty criteria does? Basically, the examiner does a patent search and the literature search. So if it is something which is hidden in literature, they would obviously in US nobody would be knowing that turmeric can heal wounds. In India, we all know about it. It's by word of mouth also it is passed on. So the initiative of TKDL was basically so that when a patent examiner searches for such things, he goes into the right sloka of the Ayurveda or the Ayush, and he can know, yes, this is a known property and it should not be given a patent. So that was a defense mechanism which the country has created. Now, when you apply for patent application which has a biological material in it, maybe you have a patent where the product is coming out of a biological product. Obviously, the plant cannot be patented, the variety cannot be patented. But you say from sugarcane, you have taken out something. So the source and geographical origin of the biological material should clearly be disclosed in the patent specification. Where did you get that material from, for which you have developed an inventive technology, which is a new and which you are going to ask for a patent. Secondly, if the biological material used in the invention is from India, permission from National Biological Authority has to be obtained by the applicant, and the same should be submitted to Patent Office before the grant of patent. Here, what I was talking to you about, that although National Biodiversity Act is not an IPR law, but because we are dealing in plant genetic resources, we have to know about the regulated access which India and all other countries have created on your plant genetic or the bioresources. So this is the crux where the IPR and the access to genetic resources meet. So how do you take access to genetic resources? Most of the patent offices will ask you whether it is legally acquired, right? So legally acquired is on mutually agreed terms, right? You create mutually agreed contractual agreement on bilateral basis and provide opportunity to providing country to negotiate a share of the benefit derived from the use of genetic resources. May specify the permitted or prohibited use of the genetic resource provided, and which can include whether or not it may be commercialized. I give you an example. Suppose as a researcher in genetic resources, you get some material from outside, and then it is given to you, and you develop a variety out of it, which can be highly commercialized. But when you reach the mutually agreed terms, you find that on the terms of conditions of the material under which you have accessed that material, the contractual agreement says that you may use it for research, but may not commercialize it. So all your work would be wasted, right? So the point is that you have to be very careful when you access material from outside or you provide material to somebody from outside. These mutually agreed terms have to be carefully seen by you. And you can negotiate your benefit out of the use of the genetic resources or also the commercial use of the genetic resources. Related to it is the concept of prior informed consent. Now, when you want something from the other party, it should be through prior informed consent. Now, prior informed consent is that you have to provide the information concerning the purpose for which they required and the proposal for benefit sharing. The authority may consult indigenous local communities, other stakeholders, and responsible authority of the provider country can grant or refuse access. You see that prior informed consent is different from consent form. Prior informed is that you have to tell the user what is your intended use of that. I give you a small example. Suppose you are on a seashore and there is a man who is throwing stones somewhere here and there, and you go and see that stone and you find that these are pearls basically, which he has oyster shells in which there are pearls, and he's just whiling them away. And you take his consent that, okay, I'll give you 100 rupees and you give me these stone-like things. And he gladly gives you. That is consent. You can say that, yeah, I've taken the consent, but it is not an informed consent. Informed consent would be that you tell that person that what you're throwing is a pearl and I would like to take it out, polish it and sell it for about 1,000 rupees. And then you tell me how much money you would like to have, and then we can share the benefits out of it. So that is prior informed consent. So for genetic resources also, you have to tell what is the intended use. You have to clarify right from the beginning. Now, under the ITPGRFA, the multilateral system of access and benefit sharing has been implemented. You are in NBPGR and training is going on NBPGR. I'm sure somebody must have touched this topic of International Treaty for Plant Genetic Resources. Here, I would like to say that this is an outcome of the CBD, and this is one of the ways when you apply for a protection of a plant variety or protection of your genetic resource, product or process derived out of a genetic resource, you would be asked whether you have legally acquired or not. So these are the systems or the instruments which will say that, yes, I have legally acquired, either through multilateral system or through MTA or with a PIC. What happens in the case of Biological Diversity Act we were talking about? This also concerns largely with access only, and it has got nothing to do per se with IPR. Although IPR has to be taken with prior approval of NPA on any bioresource, which includes plant genetic resources, obviously. So the act, as I said, is derived from CBD. So the basic objective is conservation, sustainable use, and equitable sharing of benefits, use of genetic resources, as we have seen in the case of CBD. So how does the access is regulated? For non-Indians, the permission has to come from National Biodiversity Authority. For Indians, you have to intimate to the state biodiversity boards and the involvement of local bodies in decision-making regarding the use of biological diversity. Under the Act, there are funds at national, state, and local level for conservation and benefit sharing, and also for awarding communities for conservation and of genetic resources. IPRs, as I said, you have to take prior approval of NBA whenever you want to file an patent application or plant protection. Obviously, plant variety is not concerned in this. Exemption is international collaborative research project between institutions, including government-sponsored institutions, as such institutions in other countries. So for that, you may not have to go to the Biodiversity Act, but there is a performer which ultimately you have to have the MTA and the PIC forms in that, and then the government will accept your form, and then it will give it to you in no objection, and then you can go ahead with international collaboration. Now, what are the regulations? Basically, 4 activities of related to genetic resources are regulated under the Biodiversity Act. It comprises, I again clarify, all bioresources, not just plant genetic resources. So research of biosurvey and bioutilization for research. As we discussed earlier, for personal entities under Section 2, Indians have a different formula, different protocol, and the foreign nationals have a different protocol. For research and biosurvey and bioutilization, for commercial utilization. So everybody is regulated under this if you use for biosurvey or bioutilization. Transferring of research on biological resources to personal entities under Section 3.2. There are some exceptions like presenting a paper or something, but yes, you cannot transfer research on biological resources. One more thing, I have not written it here, but it is always in the Biodiversity Act, biological resources and information related to that. So even the information cannot be transferred without seeking the prior approval. Seeking IPR over product process based on research on any bioresource, it is also for all the person, anybody, whether national, Indian national or a foreigner, if it applies for an IPR occurring on utilizing genetic resource or bioresource occurring in India, they have to take permission of the Biodiversity Authority. But certainly breeders' right under PPBFRA are exempted because there is a different law for protection of plant varieties. So if you want to have a protection of a plant variety, then you need not go to NBA for approval. And third party transfer of access by resource, also you will have to take the permission of the biodiversity. This means that suppose I have been given permission of accessing a bioresource by the biodiversity authority, but I want to transfer that bioresource to some other person, third party. So there again, I have to take the permission of the biological national BDA under the authority. Now, this is important. I don't want to make you worrisome, but then these are the penalties if you don't obey this. Sections 3, 4, or 6, which we were talking about earlier, excess, regulated excess, punishment with imprisonment for a term which may run up to 5 years, or with fine which may extend to 10 lakh rupees, or with both, right? And offences under these acts are cognizable and non-bailable, and provisions of this act shall be in addition to the provisions in any other law relating to forest or wildlife. Because as you know, we have forest laws and wildlife laws for protection of bioresources. So they will apply along with the Biological Diversity Act provisions. So you should be very careful that you should legally acquire whatever material you acquire from excess, from wherever sources, and then proceed in a legal manner. Now we come to how IPR affected the plant genetic resources. Article 27.3b of the TRIPS is the most quoted, I think, article by all our plant sciences people, because it says plants and animals other than microorganisms and essentially biological process for production of plants or animals are biological processes. So you see, these can be excluded. So when we talked about our Indian patent law, and I gave you that provision, so more or less same thing is repeated there. So it is TRIPS compliant. Now, the second line is more important. However, members shall provide for the protection of plant varieties, either by patents or by an effective sui generis system or any combination thereof. Right? So what does it mean? That although in the first paragraph, they have excluded plants under the TRIPS agreement, which we have also excluded from patentability in our act, in the second it says that protection for plant varieties has to be provided. So either by a patent or any other effective sui generis system or any combination thereof. Now, sui generis is because we did not want the patent, we have not opted for the patent, so it was an effective sui generis. At that time, there was already a system of UPOV existing when these provisions were being made. And many of the countries joined or they wanted to join UPOV. But our government or the national Indian government thought that we will create our own sui generis system. Why and how, we will talk after this. Now, this is what I was talking about. UPOV, an International Union for Protection of Plant Varieties. It's a French name, difficult to pronounce by me. So this is the easier way. And it's an intergovernmental organization with headquarters in Geneva. And established in 61 and entered into 68, 72, 78, 91. And the 78 Act in 91. And then forced into 1980. So you see, by that time, these laws for protecting varieties were being formed up by different countries due to their obligation to the TRIPS and the WTO. UPOV was already there. And many people wanted that the UPOV should be the option for people to ensure that the member states acknowledge the achievements of breeders and UPOV varieties. Obviously, that was the purpose. And they had different criteria, which was distinct from existing variety, sufficiently homogeneous, stable, and new. That means they have not been commercialized by the reference date of the application of protection. So with some criteria, the purpose was to protect the breeders, new plant varieties, to make some intellectual property protection for the breeders of new varieties. What was the advantage of UPOV? Why people wanted to go into UPOV? Because it promotes international harmonization and cooperation, mainly between its member states. Suppose you have a variety protected under one UPOV country, and then you want to go where there is another country which is also a member of UPOV. So you know the rules and protocols and procedures, and many a times the reports can be exchanged. So international trade required uniform or at least mutually compatible rules would be there. Now, there is a great degree of harmony and in practical operation of the protection system. Basic concepts would be there. The specific activities undertaken with UPOB leading to recommendations and model agreements and forms would be there. And the forum for exchange of views and sharing experiences would be there among a large group of countries. But yes, the India, even though seeing all these advantages, as a country preferred not to join UPOB, but have its own Protection of Plant Varieties and Farmers' Rights Act. The Act established the Protection of Plant Varieties and Farmers' Rights Authority and Plant Variety Registry. Reason mainly being that there were two reasons. Number one, in India, the protection was there, give the large component of intellectual property in the existing varieties was of the public sector, right? And secondly, in India, the farmers were also having their own traditional way of seed development and exchange, which we did not want to disturb. So we wanted to have the farmers' rights incorporated into the Act, which the UPOB did not have. So therefore, we created the Protection of Plant Varieties and Farmers' Rights Authority. So what is the scope? It protects any plant species that grows in the country that can be produced or commercialized for any purpose of cultivation. But then it notifies the species in Gazette of India. At present, I think more than 160 plant species have already been notified. And then under those species, when they open up for registration, you can put in your application for the protection of plant variety. Wild relatives or conserved traditional varieties or land races by farmers of any species, if not notified, can be protected. You see, this is what our wealth with the farmers was. Therefore, we wanted a new type of an act as compared to UPOB and have some provisions for protecting wild relatives or conserved traditional varieties or land races by farmers. What are the rights which are confirmed to the right holder, the person who protects? Like any other IPR, like patent we were talking about, it is an excluding right that it excludes anybody to produce, sell, market, distribute, import or export of the variety without the authorized use of the right holder. So this is the right confirmed that without if you have a protected plant variety, nobody can produce, sell, market, distribute, import or export without your permission. Now there is, as I said, specific period is there. So yes, here also 18 years from date of registration for trees and vines, and then 15 for other crops from the date of registration. And extant unnotified varieties, we will talk about it. It is also 15 years from date of notification by the Seed Act. And the regulations came in 2006. You can file application at the branch offices, which are given there. For update, you may visit the site of PPVFRA. But then other states not covered above, application form may be filed in the head office at New Delhi, which is very near to my office. Now, what are the varieties recognized as per the IPR? Means you can have a intellectual property protection of a new variety or an extant variety, which has been notified under the Seed Act, on an extant variety, which is a farmer's variety existing, or a variety of common knowledge existing, or an essentially derived variety, or a farmer's variety, right? So these are the different type of varieties which can be protected under the Plant Variety and Farmers' Rights Act, PPVFRA. Farmer variety would mean a variety which has been traditionally cultivated and evolved by the farmer in their fields, or is a wild relative or land race of variety about which the farmers possess the common knowledge. So even these varieties as farmers varieties can be protected under the PPVFRA. new variety. New variety has a criteria of novelty, obviously. Akin to the patenting, that should not be registered under the Act if it is conforms to novelty, distinctiveness, uniformity, and stability. Now, novelty, if at the date of filing of application, the propagating material or harvested material has been sold for a period of less than 1 year in India, or trees and wine 6 years, and other cases 4 years outside. Now, you see a slight different from the patent. In patent, there was a prior art search for novelty, right? That this information should not exist anywhere. There should not be a disclosure of the information. But here the novelty means it is something which is a commercial novelty. Means it is not available to the farmers already in a commercial scale. Therefore, it is a new variety, and therefore you need protection. So you can have a new variety protected under the PPBFR Act. Extant variety. Here the novelty criteria won't count. Why? Because we know that it is already existing in the market and it has been notified under Section 5 of the Seed Act 66. Okay? So therefore, the novelty criteria in this case would not count. And there is a time limit for registration of varieties of various crops fixed by the authority, which can be checked from the side. Means, suppose they notify a particular genre, they will also notify the time limit under which you have to file for the extant varieties if you have in that particular crop. Same for the variety of common knowledge, then because it's a variety of common knowledge, there will be no criteria for novelty. It is not released and notified under Seeds Act, but is well documented through publication and is capable of satisfying the definition of variety. It is under cultivation or marketing during the time of filling of application. The two representative seed of the variety should be available at the time of filing the application, or sold or otherwise disposed of in India more than 1 year but less than 15 years prior to the date of filing of application. And so this is what the variety of common knowledge is there. Then the EDV. EDV is predominantly derived from an initial variety, from a variety that is predominantly derived from such initial variety. Basically, do not go into the details if it confuses you. It is largely meant for protecting varieties which are developed from an initial variety through transgenic approach or a backcross derivative, mutant, chimera, somaclonian. They can all come under the EDV. Now, what is the dust test? We were talking about dust test. So these are the basic requirements. Although dust is mostly said, but it includes novelty, which we talked earlier. And distinctiveness is distinguishable by at least one essential characteristic from a variety, which is a matter of common knowledge. So this will be tested against the different checks. And if it is distinct, then certainly your variety would be considered for. These are all grow out tests, for extant 2 locations for 1 crop seasons, and for new varieties, 2 locations for 2 crop seasons they are grown to check for the distinctiveness, uniformity and stability. And for EDV, it is decided by the authority on case-to-case basis. So sufficiently uniform, obviously, in essential characteristics, and stable characters should be there even after repeated propagation. And denomination, this is important. You also have to give a denomination which gets protected. Suppose you have a variety and you have to give it a name, and that name also gets protected along with the variety which you are protecting. So denomination is important, which you have to provide. Now, distinctiveness we have talked about, that it is a grow test, and against proper checks, it is checked whether it is distinct or not. And then acceptable level of variance are there for uniformity. Off types and variants are described in the rules and the for the dust testing for each and every crop. So for tomato, rice, millets, I've given some examples. If more than the above observed, the company representative would be called to the site immediately by the registrar for self-assessment, and a consensus on uniformity of the material can be developed over there. This largely is the process if you want to protect your newly created plant variety under the PPVFR Act. Once you submit the application, the PVP examiner would verify the application for completeness. If it is complete, obviously it will go further along with the fees and the seed. You have to deposit the fees, we'll come to that later, and a designated amount of seed, which is told as per the rules and laws. You have to provide the seed also. So if it is not complete, then notice of completion would be given, and then the amended application with all its details would be published in the Plant Variety Journal of India. So then there is a provision for opposition. So if there is some opposition, then you will be given opposition notice to the applicant. There will be a fee, and in 1 month you can counter the opposition. 2 months time is given, and then the serving of the counter statement to the opposing party would be given, who has raised opposition with you, and then the hearing if required. The next is plea rejected on both sides. On one side, it should be plea of the opposition is rejected, and plea if accepted, then the proposal would be rejected. And once there is no opposition, then it goes for the dust examination. And when it clears the dust examination, then the issue of certificate of registration, and will be published in the Plant Variety Journal as a protected variety and entered into the National Registry of Plant Varieties. So after that, you have to pay the annual fee. There can be a provision of compulsory license, and then there's to be a benefit sharing and revocation and surrender. Now, we have talked about the rights to sell, produce, market, import, export, right? But then what are the exceptions to these rights? One is the researcher's right. Means anybody can conduct experiment or research on any protected variety. That is not called as infringement. And you can use as the initial source of variety for the purpose of creating other varieties, provided that the authorization of the breeder of the registered variety is required, where the repeated use of such variety as a parental line is necessary for commercial production of such newly developed variety. Now, there are farmers' rights also. The Act specifically recognizes the farmer as a breeder, as a cultivator or a consumer, and as a conserver of genetic resources. So, breeder because new varieties and extant varieties, including farmers' varieties, are eligible for registration. So farmer can even have a, if he's a progressive farmer, a new variety, register it as a new variety, or extant variety, as we said, already existing in the market, or if a farmer's variety in the form of a land race or a wild relative, the farmer can protect. That is the farmer's rights one of them. Now, the second is very important. The farmer is entitled to save, use, sow, re-sow, exchange, share, or sell his farm produce, including seed of a variety protected under the Act. This is one of the major Act provisions which the UPOP does not agree to, and many of the people don't agree, private people. But yes, it is to save the traditional seed trading system in our country. The farmer can save, use, sow of a farm produce. The only thing is he cannot brand the seed of a protected variety. Suppose it is some variety, the farmer can produce and sell it to his neighbor without labeling it as that branded variety's name. XYZ, suppose the variety is XYZ, the farmer can use, save, sow, or resow, or exchange or share the variety seed of XYZ, but while giving it to other people, he cannot market it as an XYZ seed and brand it. That is the only provision which stops it. The registered breeders must disclose expected performance under others. This is another, like consumer, as I said. Farmer is a consumer also. So any breeder who has registered a variety must disclose the expected performance under given conditions. And suppose that performance is not met, then farmers can claim compensation. So farmer is a consumer here. Protection of innocent infringement means right not to be deemed infringed by a farmer who at the time of such infringement was not aware of the existence of such right. What does this mean? That if a farmer is growing a protected variety and then he is not aware that it's a protected variety and I'm resowing it or something, he can do that, but it cannot be deemed as an infringement because of the such right was not known to him. Disclosure in the application information regarding use of genetic material by any tribal or rural families in the breeding or development varieties. So in the application form, you have to mention there, and also the communities can file in notified center claim for compensation if such community has contributed significantly to the evolution of the registered variety, and allows compulsory licensing. Compulsory licensing is, suppose you have protected a variety and you are not providing it to people due to unreasonable price or levying unreasonable price on that seed to the farmers, the government can go for compulsory licensing it and license it to other people to produce that variety and make it available to the farmers. This is the publication which is brought out by PPBFR side, and you should, if you are a breeder or a genetic resources person, should make it a point to read it like a patent database for two things. One thing is you would know that if your material has been used by somebody else, and then you can oppose it for protection. And the other is that you can also see what are the material being protected and you'll be aware of that. These are the schedule of fees. I think you should check the websites regularly when you have to go for these acts under the PPBFR, because they can change at any time. But the point I want to make is that it's not a free lunch. If you want to protect your IPR, it is always a cost attached to it. So be prepared for that. Now, nullity actions under PPBFR, when can your claim be nulled? Then this till the registration of a variety, we talked about opposition. Somebody can oppose and say that this is not correct. You are giving it a wrong information. And after registration, it is revocation. It can be revoked, or through infringement if somebody is doing infringement. Opposition, entitlement, that somebody can say that I'm opposing this protection of plant variety material because this person is not entitled, is not the rightful owner of the intellectual property which has been claimed there. Then it is a non-registrable variety, means that it is failing against the dust test or something like that. It is non-registrable. There has been a problem in that. And against public interest and adverse effect on environment, these things are there. Revocation can be there if you provide incorrect information in the application, and or if it comes to the notice of the authority that it has been granted to a person who is not eligible for protection, the ownership lies with somebody else, and the requisite information has not been provided. No alternative denomination provided by the breeder. What happens is, suppose you provide a denomination, right? XYZ, and the authority finds that there is already a denomination XYZ, and then they want you to change their denomination and provide an alternative denomination, and you don't provide anything, then they can revoke your certificate. So if you don't provide necessary seed propagating material to compulsory license, then your registration can be revoked. Breeder not complied with any provision, obviously, it will be there, or the directions or grant against public interest. Such self-explanatory. So under these conditions, the PPVFR certificate, which has been given to you as an IPR right, can be revoked. There are criminal offenses here also. If you're wrong, representing a variety as registered, there are punishments, and you can see here. So with this, I would like to thank you all for your listening, and
Thank you very much, sir.
questions we will be ready.
Now, I request our participants, with your permission, sir, take a few questions, sir, from
Sure.
participant. Sir, there are one question in chat box. Sir, in contract farming, farmer can use the seed next year for cultivation and exchange the seed to other farmers.
As per the PPVFR Act, farmer can do that, right? Because he can sow, resow, and exchange material with other farmers of a protected variety. But he has said under contract farming, right? So what happens is, once I am entering into a contract farming, means that I am entering into a contract with that particular private person. In that contract, he writes that I'm giving you this seed only for this season. So in the next season, you will not be able to do it. And he puts all those conditions in that contract, and that the farmer signs. Therefore, that confusion comes that why a farmer cannot do it. So although if there is a provision in the law, that contract, I think the government of India should look into it, that such provisions should not be a part of the law, because the law does not permit that provision. But yes, that problem is coming. That's a contract between the farmer and the company, which prohibits him from doing those acts. As per PPVFR Act, he can do it.
Okay. Sir, another question from Dr. Vikas Pawar. He is asking, is State Agriculture University can apply for geographical indication or not? GI.
Geographical indication, as I said, is given to the growers or the producers or the sellers. It's a collective mark, you know.
Yes.
So university can facilitate that. You will have to create a group of people which are producers of that particular geographical indication product, right? So that the benefit goes to all, right? It is not a one-man thing. So university can itself not register it. University can facilitate in filing of the GI for different products of their vicinity, which they identify are unique and or their characteristics because of that particular geographical region. So that is the answer. But university is not an applicant. Applicant would be the group of people who are producing or nurturing that geographical indication.
Okay, thank you, sir. And another question from Dr. Amar Kant. He's asking, sir, where boundary is not restrict, say for Nepal and West Bengal, and some genetic material movement is to there. What are the norms and rules for protection in such situation?
No, that is a law and order situation. drugs are coming from across the border,
Yes.
right? So that is not a problem of genetic resources. What we are talking is legal access, how you can have legal access. If somebody goes beyond that, that's an illegal act. And it has to be dealt with appropriately.
Sir, one more question.
But one thing I can say here, you must be having a lecture on that. Transboundary disease transmission is becoming a very important topic nowadays. And there are efforts going on for transboundary disease protection because it happens inadvertently, right? So there are a lot of progress in that side. I think one of the lectures must be on quarantine. They will deal with that. And you should ask this.
Yes, sir. We had a lecture, sir. Dr. S.C. Dubey and Dr. Celia Chalam, they have delivered the lecture and they covered this aspect, sir.
Yes.
Sir, another question is from Dr. Devrajan. He's asking, after the protection of period of 15 years, what would be the status of the registered variety?
It will be in public domain. Anybody can use it without any restriction. That's what I'm saying. For patents also, there are many people who keep a watch on the patents which are going to expire. Some good product, people keep a watch on the patents which are going to expire, and 2-3 years before its expiry, they start making that thing, right? And as soon as the patent expires, they put it in the market. So after the all generic medicines which you have, they are all where the patents has expired. Similarly, in plant varieties, you can, if there is a plant variety and you want to use it recurringly for your particular variety, wait till the 15th year, do all the work. After 15 years, the day it's over, release your variety, nobody can stop.
Thank you, sir. Another question from Dr. Reena Mehra. She's asking, if some variety released in different country, another country like Bangladesh, and if we take those variety at our country, would it be allowed to evaluate and released as a variety?
If it is a protected variety in Bangladesh, right? That variety is protected in Bangladesh. So technically, as I said, geographical boundaries are there. You can do that. But if you export that variety to Bangladesh, you will be in prison. Because you cannot export that variety. You don't have any license to do that. But, or if that person is also protected in India, then it becomes a protected variety in India also. But otherwise, anything which is protected, IPR protected in a country, the jurisdiction is that country. There is nothing like a global thing. But you will be playing on, number one, illegal work, and number two, very risky work, because the day somebody sees you that, and it can be proved that you have given material to Bangladesh or somewhere, and then you can be taken to the court. So better not do it. Take a license, and I think if you, it has a market in India, that person will be too willing to give you the license for export and import, and do it.
Thank you very much, sir. Dear participant, any other question? If
IPR is basically, it does not stop you from working. If you have that IPR in mindset, what happens is that you know the right way to do things. I give you an example. People generally get flabbergasted when we talk about IPR and this and that and get confused. IPR is like any other property. It's a property which has emanated from your mind. That's the only thing. You have a house, a property. What do you do? Once you want to buy a house, you go for all legal purposes. No? You ask whether this is original, this person is the original owner or not. Is it not? Whether if I take this house, there will be no obligation on me that tomorrow I have to pay the rent or something or this and that. Once you acquire it legally, it becomes your house. And when you have that property, you protect it. Similarly, intellectual property, you protect it. How you protect it? आप उसमें लॉक लगाएंगे, ना? आप क्या करेंगे कि आप उसके अंदर एक चौकीदार भी बिठा देंगे अगर आपको लगता है कुछ ये गड़बड़ है, कहीं डबल डोर लगाएंगे, सीसीटीवी कैमरा लगाएंगे. Same with intellectual property. You can have a patent on it, right? And once the patent is there, then you find that this can be easily copied by other people once I bring it into the market. So you have a design protection also. ठीक है ना, एक चौकीदार भी बिठा दिया आपने कि कम से कम बनाएगा तो इस डिजाइन का जो मोस्ट एफिशिएंट डिजाइन है 4-5 ये तो नहीं बना सकता वो. Once that is there, you create a trademark of yours और उसको पॉपुलराइज करिए. नाइके का ही जूता खरीदोगे ना, दूसरा तो नहीं खरीदोगे. तो आपने अगर बार अपना ट्रेडमार्क बना के उसको प्रसिद्ध कर दिया तो मैं कितना ही कॉपी करूं आपकी चीज को, मैं वहां तक नहीं पहुंच सकता. फिर आप उसका एक जिंगल बनाएंगे, एडवर्टाइजमेंट देंगे टीवी पे, उस पर कॉपीराइट लेंगे. तो जो वर्ड्स आपने यूज करे हैं अपने प्रोडक्ट को डिफाइन करने के लिए बार बार लगातार, है ना, वो कोई और यूज नहीं कर सकता. सबसे सफेद चौंक गए. These are all protected. Nobody else can use it. So this is your property, and different ways you have protected that property. Right? Think it that way. Then it will be much easier. पहली चीज IPR आपको ओनरशिप देता है कि भाई जैसे मकान की रजिस्ट्री होती है ना आपके नाम पे, जब आप कोई IPR ले लेते हैं, पेटेंट या प्लांट वैरायटी, you become the owner of that particular. So everybody knows कि he is the inventor, इन्होंने ये चीज बनाई. दूसरी चीज वो आपको विजिबिलिटी देता है.
गेंदे की सीडलिंग है क्या? अब नहीं है ना?
हाँ, there is some confusion.
Please mute.
I give you another example. We have time for 5-10 minutes. So I'm stopping your lunch anyway. There was an institute which was dealing in poultry when we were just getting into the IPR business in ICR. What happened that we talked about that they used.
Oh, sorry. It's muted.
Sir, your voice is muted, sir. Please unmute, sir.
So they used to have the chicken being sold at the farm gate, right? It was a good chicken. Then there was a local market vendor who used to take that chicken and sell it at a premium, because that was a better chicken. And then they would say, try to negotiate with him, please negotiate and give us something. He would not give. फिर क्या कर सकते हैं, अपने इंस्टीट्यूट के ज्यादा से ज्यादा एक आदमी को एक देंगे. वो बड़ा आदमी हो 10 आदमी लगा देगा पीछे, 10 आदमी खड़े हो जाएंगे लेने के लिए. So it was not working out. Then we decided that let's take a trademark on it. Right? We can't patent it. We took trademark. And the trademark was Vanyaraj and Gram Priya. Many of you must be knowing this name now. Is it not? Vanyaraj and Gram Priya were taken as trademarks. Now what happens is that man can take it, take 10 or 15 from there, sell it at a premium also, but cannot say that this is Gram Priya or Vanyaraj. The day he says that, he will be behind bars. So he has to come on negotiation table that I label ki I am selling Vanyaraj and Gram Priya, please provide me this. So that is ownership that he has established. Second is visibility. After that came the NATP project on backyard poultry, right? And these breeds performed exceedingly well from east to west and north to south. Atak to Katak, everywhere it was a very good breed. What would have happened suppose these trademarks were not there? Say after 5, 6, 8, 10 generations, someone you have given it as a PDP 50 not 503 or something like that. And then after 5 years, 6 years, somebody would have named it Moti, Hira, Kanti, whatever it is, in different parts of the country, and people would have asked, what has ICR done? You keep saying, but now you proudly say One Raj Grand Prix, and now I heard that there's a lot of series of poultry breeds which they have taken off. These are our breeds. So it gives you visibility. And third, if you are able to commercialize it, it gives you revenue also. My point is, we are in public sector. Revenue is a relatively lower priority for us, but certainly now it is becoming a big priority. And you should aim that whatever research you are doing should generate some revenue out of what you are doing. And IPR brings that for you. You protect it. You gives you much better negotiating power when you want to license your technology or transfer your technology to somebody. Therefore, IPR becomes very important in that case. And my motto would be that you can give it for free also, because regulations and IPR, please be clear on these two. Regulations are different. Regulations you have to follow. It is obligatory. IPR is not obligatory. You don't want to take patent, don't take patent. But if you want to access, it has to be in the right manner. You can't say the मैंने तो commercialize करना नहीं था इसलिए मैंने access ऐसे ही कर लिया. No, that is a regulation. I'll give you an example. Suppose somebody invents a motor engine which works in water. You only need water, boiled water, and the car can go. You'll get a patent. It's novel, it's utility is there, and inventive step is there. Easily you'll get a patent. But can you put it in your car and start driving that? No. You have to have a roadworthiness certificate, which is given by Budni. Once you get that, you can put it and start in your car. Can you drive like that? No, without a proper license, driving license, you cannot drive. And even if you have a driving license, you cannot drink and drive. These are regulations. You may have a very good IPR, world's best selling IPR, and a car engine running on water, but you can't forego the regulations, which a plant genetic resources institutes we are all dealing with should have in mind. I tell you because of one thing. We all sell achar, papad, and these type of things on our counters. They are regulated. Look for the regulations. FSSAI approvals are there or not before you sell it. Right? Once you have FSSAI approval, I think your technology transfer would also be much faster. The person who is trying to take your technology would be very convinced that yes, it is an FSSAI approved product, so let me have it. If you want to sell your microbial combination, all the regulatory tests you have to do. Right? So my point is that IPR, okay, it defines only your boundaries and your things, but regulations are equally important. And keep track of the regulations in whatever field you are working and whenever you are working. I think I've taken a lot of time and stopping you from lunch. So thank you very much. If anything else is there, otherwise we can call it in.
So any participant, if they have further any question, so they can contact sir, they can write the email to the sir, and sir may answer for their queries. So thank you very much, sir, for your excellent talk about the burning topics of IPR to make the participants aware and clear about these issues, how they can protect their variety and protect their properties. So on behalf of NBPGR and on behalf of Training Organizing Committee, sir, I would like to thank for your sir wonderful talk and very important talk and accepting our invitation, sir. So thank you very much, sir.
Thank you. Thank you very much.
Thank you.
Thank you,
sir.
sir.
Namaskar, sir.
Thank you, sir. Thank you very much for joining with us.
So
Okay, madam.
yeah, Subhas, 2:30.
2:30, madam. आप थोड़ा देख लीजिएगा ना.
So all the participants are requested to join by 2:25. So for a
Another-
lecture at
Next
2:30.
lecture is by Dr. Sushil Kumar, sir.
Okay.
This is a processing of released varieties and management of millets genetic resources.
Yeah, for the breeder, this is an important lecture because for releasing the variety, they have to get the IC number of the variety. So how to get the IC number and how to submit in the gene bank. So those all process will be dealt by sir, the all procedures. Good afternoon, sir. Pandey, sir. Good afternoon.
जी मैडम नमस्कार।
नमस्कार सर, नमस्कार सर। So, दो तीन मिनट में सर स्टार्ट करेंगे, अभी कुछ लोग और ज्वाइन कर रहे हैं
नो प्रॉब्लम।
तो
ठीक।
So, sir, in the meantime, Pandey sir, I would like to tell about the participants. As now, most of them have joined.
Yes, madam, please go ahead.
So, sir, here we have more than 80 participants, sir, and they mainly, their discipline are mainly, sir, plant breeding and genetics from different background. They are working on different crops like rice, wheat, then vegetables, pulses, oil seeds, even from tree and forage also means they are there. And mainly they are from sir SAUs, sir, State Agriculture University, 59 of them. And 45 of them are sir assistant professor. Most of them are assistant professor. And then we have associate professor, professor rank also. Some of them are there. So there is a good means diverse group, sir. Means as our training is about PGR. So here also good diversity in the group. So good afternoon, everyone, dear participants. So today, first I would welcome today's speaker, Dr. Sushil Pandey, sir. So I'm sharing my screen to introduce, sir. So hope it is visible.
Yes,
So sir is a principal scientist in Division of Germplasm Conservation at NBPGR. He has obtained his master's and PhD degree from Division of Seed Science and Technology from IARA New Delhi in 96 and 2000 respectively. Sir has joined ICAR in 2000 at National Research Center for Soybean Indore, and then he moved to IARA New Delhi in Division of Seed Science and Technology. After that, he joined as a senior scientist through direct selection, and he moved to ICR NBPGR New Delhi in 2009. And since 2015, sir is principal scientist here. So regarding sir's achievements, so sir has developed hybrid rice seed production technology under North Indian condition. He has, to his credit, about development of hybrid tomato seed production technology under protected cultivation under North Indian condition. In addition to that, he has developed guidelines for improving quality and effectiveness of seed provided in emergency operations of FAO across the world. So sir has been deputed to FAO several times, and that is his great contribution for developing guidelines. Then he has developed a methodology for breaking seed dormancy in different vegetable crops. Then sir has also developed PGR interactive software and some registration number is given to that software. And to his credit, more than 60 research papers are there. And sir is also fellow of Indian Society of Seed Technology. So sir is in Division of Germ Plant Conservation, and so today sir will deal about how to register the variety in the NBPGR, what are the guidelines for that. And then he is the expert of millet genetic resources also. So we'll deal about millets also. And here sir is looking after a big platform, consortium research platform, agro biodiversity. So he is doing the great role in coordination of that program, is running over almost more than 30 centers across the countries in various crop for characterization, evaluation as far as pre-breeding products. So sir has great role in running of that particular project, because that is for various crops, then fishes, animal, it is for all the genetic resources. So thank you very much, sir, for accepting our invitation. And I am stopping my slide, sir. So now you can share your slide and deliver your talk.
Thank you so much, madam. Sometimes it sounds good listening about yourself. So thank you.
Thank you, sir.
With your kind permission, I will share my slide.
Sure, sir, please, sir.
1 second, I will first open. Share screen. So is
Yes,
it
sir,
okay?
Yes, sir. Yes, sir. It's okay.
I will
sir.
start now.
Okay,
So,
sir.
thank you and good afternoon, participants. So I will be dealing with the processing of release variety and also the management of millet genetic resources. So already I think you had a lecture on millets, Dr. Tara Satyavati covered. So millets are not new crop for you all, and we have major millets, that is pearl millet, sorghum, finger millet, and small millets we have foxtail, barnyard, proso, kodo, and little millet. In fact, these are very old grains. In before independence, almost 25% of Indians, this was the staple food. However, now over the year with the adoption of wheat and rice, now only 6% of the population, Indian population, it is taken as staple food. So why there is renewed interest in millets? Mostly because it has been recognized that millets, they are highly nutritious. They have high calcium, iron, potassium, magnesium, and zinc, besides other essential molecules such as vitamins, amino acids, and fatty acids. Then again, millets, they have inbuilt tolerance to water stress and high temperatures. And their morphophysiological, molecular, and biochemical characters make them more tolerant to water stress and high temperatures. And again, being a C4 crop, they have higher potential in utilizing the atmospheric CO2 and better photosynthesis per unit of water which is being used. So these are the main reasons why there is again renewed interest in millets, especially because of changing climate. So one thing I would like to appreciate about Indian government is that since they wanted to promote millets, they have stepwise they have taken different initiatives. Like in 2018, the government had notified nutri-cereals. So all the 8 millets, along with buckwheat and amaranthus, they were recognized, notified as Nutri cereals on 10th April 2018. Then again, 2018 was again declared by government as National Year of Millets to create awareness about the good attributes of the millets. And then now, presently, we are having International Year of Millets, which has been accepted by United Nations General Assembly, and almost 70 countries, they have adopted this, and they are also celebrating the International Day of Millets. So just briefly about telling about millets, you can see that from 1950 onwards to 2020, different millet crops, how they have reduced. The area has reduced continuously over the years, and more drastic reduction has taken place after 1990. So this is sorghum, this again pearl millet. The total reduction in area may not be that much as drastic as it is in sorghum, but again in finger millet and small millet, the reduction is continuing, and there is 60% decline of area from 34.8 million hectare to 13.6 million hectare now. So now coming to the contribution of different millet crops at international level. So kodo millet, little millet, barnyard millet, they are completely 100% area percent share is in India only. Whereas in case of finger and pearl millet, 50% of the total production is from India, and remaining is from abroad. In sorghum, we contribute 7%, foxtail millet 2%, and proso millet 1%. Now coming to within India, the main regions growing sorghum, they are Karnataka, Maharashtra, and Rajasthan, whereas pearl millet is grown mostly in Maharashtra, Rajasthan, and UP. Then finger millet, more than 60% area is alone in Karnataka. And then other small millets, they are mostly grown in Madhya Pradesh and Chhattisgarh region. Now, this is the production, yield, and area under millets from 1950 to 1920, the trends. You can see the production in millets has increased, but at a lower rate till 1990. But after 1990, the production has started reducing. Similarly, when we go to yield, yield is continuously increasing. So this you can attribute to the technological intervention. That means the government research institute, research and development institutes, they have been continuing to develop good varieties. That's why there is productivity gain over the years. Although the production and area has up to 1990, again you can see there is very small reduction from 35 million hectares to only 32.8 million hectares, whereas from 1990 to 2020, more than 50% reduction is there. Like 32 million hectare, it has gone down to 15 million hectares. So this is the production, productivity, and area trends over the years, and it has been found, if you see this trend in wheat, rice, and other cereals, it has been found that the area depends mostly upon the area of a crop will depend upon the technology, that is technological advancement through improved varieties, and also the policies of the government. So in case of wheat and rice, you can see that the productivity has increased around 3.7 times, here 2.63 times. That much increase is there. Similarly, the area has also increased 2.2 times, 1.22 times, and therefore it has resulted in production increase of 8 times to 3 times in rice. This is mostly because of the government policy. One thing is that technology, that is new varieties are coming, Then area is also increasing because more and more farmers are taking up production of wheat and rice because of the policy of government, that is minimum support price. They purchase back the seeds, and that's why. In case of maize, you will see that technological advancement is there, productivity has increased, but the same way area has also increased, and therefore productivity has also. But here you will find that there is no government intervention policy because government don't purchase this on minimum support price. Still there is increase in area. So why this has taken place, although there is no policy support from the government, this has taken place because of the development of poultry industry from 1990 onwards, and there is lot of consumption of millets, and that's why area and production of maize has taken place in spite of no policy support. But in case of pearl millet, sorghum, finger millet, and small millet, although there is technological advance and increase in productivity, area has reduced continuously in all the crops. And similarly, you can see the production has also increased, except in case of pearl millet. Now, pearl millet production has not increased because of increase in area, but only because of the technological advancement. That is because of development of hybrid. So only because of productivity, this production has increased. Otherwise, there is no neither market driven forces are there to encourage pearl millet, nor government system is there. So again, this is the to summarize that in wheat, you have wheat and rice, you have technology driven increase, policy driven demand, and market driven demand. In case of maize, you have technology, but no policy, but still market driven demand is there. In pearl millet, sorghum, finger millet, and small millet, the policy and market driven demand is almost negligible. So here you can see now the government has taken up the matters very seriously, and now you can see over the last 10 years there is 238% increase in sorghum minimum support price and 167% pearl millet support price. So we expect that more and more adoption will be taking place. But today only I was reading an article by Dr. Ramesh Chand that in spite of more than 5 to 10 years of concentrated efforts by the government, still the total area under millets has not increased much. Only thing what has changed a little bit is millet, instead of being earlier consumed by the poor people, is now slowly shifting towards their consumption by middle class and rich people. So now, because as I said, that technological advance is playing an important role in saving the millet crop over the 70 years, although there was decline in area and production. So in fact, the millet, the genetic resources, they play important role in development of improved varieties. So with this, the role of NBPGR comes. And as we know that we have mandate of management and promote sustainable use of plant genetic resources, then coordinate and capacity building in PGR management, then molecular profiling of varieties of agri-horticultural crops and GM detection technology research. So these are our 10 regional stations across the country, and around 59 national active germplasm sites. This may be taking change within 6 months because due to various reasons, some of the national NAGs have been added extra and some have been abolished. So maybe within 6 months, we will be doing these exercises, and all the crop experts from different part of country, we will be inviting them for this. So in Gene Bank, we presently have total of 4,79,168 accessions, out of which Seed Gene Bank has 4,64,000 accessions. And this is the breakup of the seed accession, Seed Gene Bank. And you can see around 60,000 accessions of millet is being conserved out of 4,64,000, which forms around 13% of gene bank accessions. So this is the breakup of different millet crops. In sorghum, we have more than 25,000. Pearl millet, we have 8,743. Finger millet, we have around 11,500. Foxtail millet, 4,600. Barnyard millet, around 2,000. Kodo millet, 2,400. Little millet, 2,100. Proso millet, 1,000. So total around 60,262 accessions we have. And then within different crops, we like out of 60,262, we have 44,000 accessions which are of Indian origin, and 15,000 accessions in case which are of exotic origin. And the maximum is in case of sorghum, around more than 56%. They are of exotic origin. Otherwise, in other crops, we have very less, like in only 1% of exotic collection in case of minor millet, that is foxtail, barnyard, kodo, little, and proso. We hardly have very less numbers in case of the exotic material. That means we still have lot of germplasm to be, especially to be introduced from Africa, and maybe not directly, we may get it in future through ICRISAT. So these are the different states which contribute to the millet germplasm, which is conserved in National Gene Bank. And mostly the important states are Andhra Pradesh, Bihar, Gujarat, to some extent Himachal Pradesh, Karnataka, Madhya Pradesh, Maharashtra, Odisha, Rajasthan, Tamil Nadu, Uttar Pradesh, and Uttarakhand. So these are the important states which have from which germplasm has been conserved. And in fact, Maharashtra and Madhya Pradesh are the most important one among them. So now year-wise, if you see, this is the collection status, and you in 2001 to 2005, you will see that around 17,000, that is the first peak we got in germplasm collection. Now, this is the year when there was a NATP project, which was implemented, and NBPGR had a major role in that project. In this project, several thousands of material was collected from all over India, and that's why there is increased number in 2001 to 2005 in Gene Bank. Again, you can see another peak in 16 to 20, around 22,000 accessions. Now, this peak is because in 2011 to 2015, a lot of material from ICRISAT that was sent to Svalbard Gene Bank. So as per the policy protocols, they are required to submit a small quantity of voucher sample to NBPGR. So that voucher sample we got multiplied during different year and conserved in our gene bank during 16 to 20. That's why this second peak we got. So that means a lot of material from ICRISAT we have got. So this is the different type of biological status material which we conserve. That is land races, primitive cultivar, farmers variety, parental lines of hybrid release variety, registered germplasm. So in case of millets, you can see land races are one of the big, out of 60,000, more than 33% of the germplasm belongs to land races. And another is farmers variety. So around 30,000, that is 50% of the material either belong are either land races or farmers variety. But a large chunk is also there, that is around 24,000 material, which is not, which we don't know what is their biological status. So because a lot of passport data is also missing in many of the accessions, but that does not mean that that germplasm becomes useless. But we have to find out different ways how we can add value to this germplasm where passport data is missing, which I will tell you after some time. So this is again the graphical representation of the land races and farmers variety which are being conserved in gene bank. Now, this is that was National Gene Bank's data. Now this is the RS Paroda Gene Bank, which is at ICRISAT. There they have another set of sorghum, 42,000 pearl millet, 24,000 finger millet, and all around 80,000 germplasm is being conserved. So you can see that in India, we have around 25,000 sorghum germplasm conserved with us, whereas they have 42,000. So still there is a lot of material which we can retrieve from ICRISAT, and we are continuously working on this so that we can get this material from them. Then again, pearl millet is another important crop. We have only around 8,500 accessions with that, whereas pearl millet, ICRISAT has 24,000. So still around 15,000 accessions we have to get from them. And in finger millet, foxtail millet, they have sufficient, some small quantity, but almost nothing is present in our national gene bank. So over the years, we are working so that we can get these. It is not that easy, but still we are trying, and hopefully in the coming years, we will be able to get a lot of these materials back from ICRISAT. So now one thing which I was telling that since there can be some material where we do passport data is missing. And second thing is that even if material is there with passport data, if we don't have the proper characterization and evaluation data, that material may not be very useful to the breeders for crop improvement program. So in order to have better utilization of the plant genetic resources conserved in the National Gene Bank, NBPGR has started a mega ICR funded program on CRP on agrobiodiversity, and presently we are having the third phase. The first phase was from 2014 to 17. Now we had second phase from 17 to 21, and third phase from 21 to 26, and almost 50 centers are there, plant centers. We also have animal, fish, microbe, and animal genetic resources also covered in this. So in plant genetic resources, we have three main activities. First is multiplication and characterization of germplasm. Second is trait-specific evaluation for use by the breeders. And third is trait discovery and pre-breeding in selected crops, that is in wheat, chickpea, and okra. So this is the program which we have been taking since last 10 years. And this will add a lot of value to the conserved germplasm so that it can be directly used by the breeders in their crop improvement programs. And in over the years, you can see around 22,000 accessions of sorghum have been characterized in different years. Then foxtail millet, around 4,893 accessions. Barnyard millet, 2,114. Finger millet, around 8,824. So till today, out of 60,000, around 37,881 accessions have already been characterized under CRP on agrobiodiversity. And these are some of the sorghum diversity during the characterization present in our germplasm. Similarly, here is the finger millet diversity. Now, one thing which, as I told, 2023 is International Year of Millet, and NBPGR is taking up characterization program of 25,000. That is entire set of sorghum, germplasm. Their characterization is being done at Agricultural Research Station, Washem, which comes under Dr. PDKV University, Akola. This program we are implementing in collaboration with IIM Hyderabad, and already to 24,950 accessions along with checks. We have 5 checks. They are being sown in Agriculture Research Station, Washem, and presently already flowering stage is going on, and maybe most probably on March 13th, we will be organizing a mega field day where we will be inviting breeders from all over the country to visit that site. That is actually the country's biggest character. Before this, we had 21,500 accessions of wheat, which were characterized in 2011, 12 and 13. Now this is additional 3,000-4,000 accessions. So already this, and these are the 26 traits for which the descriptor states for which the characterization is being done at ARS Washem. The purpose is to develop the core set. So because suppose somebody asked the diversity from the germplasm, 25,000 germplasm nobody can use. So there is a way of identifying 10% of that germplasm. So maybe around 2,500 germplasm can represent the complete set. And then again, further mini core will be developed, which will constitute only 250 germplasm accessions, and thus it will enhance the utilization program over the years. So this sowing has already been done in November, and now you can see the growth has already taken place, and already data is being recorded for all the 25 characters. This is the status of data as on 10th February. So germination and planting, plant vigor, almost 100% data is complete, whereas other traits, the data is being so around 34% to 100%. These are the traits where then later on seed characters will be adding up. So again, with opportunity in this lecture, I inform you all that on 13th March we will be organizing a mega field day, and whoever is interested, then they can come visit there. So in addition to characterization, we are also doing trait specific evaluation, that is screening of finger millet germplasm for blast resistance at 3 locations: Vizianagaram, Mandya and Almora, under CRP on agrobiodiversity for leaf blast, neck blast and finger blast. This is the experiment at Vizianagaram. This one is at Mandya. So this is the value addition in so that this through trait specific evaluation, we can identify a good number of germplasm and get them registered in through NBPGR. So at present, we have around 146 germplasm accessions of different millets which have been registered, out of which I just wanted to inform that more than around 50% accessions have been registered in 2013, or after 2013 only, after the initiation of CRP-AB program. And moreover, out of 38 small millet germplasm, more than 92%, that means only very less, 3 accessions of small millets were registered before 2013. So we encourage the registration program under CRP agrobiodiversity, and now I'm very happy to inform that this has very positive impact. And this registered germplasm, you can all the crops you can find from NBPDR or net site, where you can go to PGR databases, and inventory of registered crop germplasm will be there. From there, you can get the traits of all the registered germplasm of different crops. So just I'm telling about some of the millets which have been registered for high protein, gray mold resistance, drought tolerance, then blast resistant, grain smut resistant, like that, high oil content. So these are the different traits for which we have already registered the genetic stocks. So with that, I will switch over to the other part, that is processing of release variety, because as Dr. Jyoti told most of you, they are working, you are working in different crops, breeding of various, and at different stages, you have to develop variety and get it notified. So before notification, it is compulsory that you have to submit the seeds at NBPGR and get IC number. So just to facilitate that you people don't face any problem, we keep on covering this topic so that everybody knows what is the procedure involved in getting IC number for the release variety. So for normal germplasm, the seed material is submitted to Head Division of Plant Exploration and Germplasm Collection at NBPGR, along with this passport data, collector ID, IC number, crop name. So this is for the normal set of germplasm, which you collect through explorations from different part of the country. This is the pro forma in which you have to submit. But for in case of conservation of release variety for notification, it is requested that, because it is the material is directly processed by Division of Germplasm Conservation for issuing IC numbers to release variety, the material can be submitted directly to director or to head of the conservation division. Along with the material, it is requested that you should submit the identification proposal of the variety. This is very important. Many people, they give the passport data in the table which I shown for normal germplasm. That is not required. We need the identification proposal of the variety, because that will help us in getting the important characters of the variety which we put in the passport data while generating the IC number. Then workshop recommendation is very important in case of release variety, because no variety, every variety, it has to go to a workshop where it will be discussed, the results will be discussed, and then the variety is recommended for release. So it is important that the workshop recommendation copy is also submitted along with the identification proposal. Then you are required to submit the fresh and untreated seed of varieties, hybrid and parental lines in sufficient quantity. That is, in case of self-pollinated crop, we have 2000 seeds and 4000 seeds in case of cross-pollinated crops. Now, in case of hybrids, CMS, maintainer and restorer lines, all the three lines have to be submitted in case of CGMS system. Many times the restorer lines are not being submitted. Please note that the purpose of conserving the release variety in gene bank is that it can be made available for the posterity. If you don't have submitted restorer line, if you want, that means maintainer line, sorry. Normally, earlier people don't used to give maintainer line, only CMS and restorer line. So if we have to multiply this CMS, we never had this maintainer line with us, and therefore it cannot be used for posterity. That's why Now we have made it compulsory that all the 3 lines in CGMS system has to be submitted. Then in case of 2-line system, male and female line both have to be submitted. Then in case of hand emasculation and pollinated hybrids, both the parents have to be submitted. And in case of maize, because sometimes more than 2 and 3 parents are involved, all inbred lines of the maize have to be submitted. Now, in case of vegetatively propagated crops, along with the information which I showed in the previous slide, a deposition certificate from NAGs, that is National Active Germplasm Sites, have to be submitted. Because the vegetative propagules, we don't conserve in our gene bank. So the vegetative propagules for every vegetatively propagated crop, the concerned NAGs have been identified. You have to submit the propagules there. They will give a deposition certificate to you. Along with that deposition certificate and other information, you have to submit the proposal to us for IC number allocation. After we receive the seed and along with all the information, we send it for seed health testing, for quarantine testing. And normally this is the for allotting IC number, we after quarantine testing, we go for seed moisture testing and drying, after which seed germination and viability testing is being done. And then IC number allocation is done for notification. Now, germination testing report, many times people, they submit germination testing report from different labs. That is not required, because in any case, either you submit the germination testing report or not, we have our own testing procedure, and therefore you don't need to. Now, in seed health testing, as you know, there are we don't reject the seeds if they are infected with in case of legumes, hidden infestation is there. That is, insects, they feed inside the seed. So through X-ray radiography, we can see so many seeds are infected from inside. So when large quantity of seeds are infested, then we are helpless. But otherwise, if we find that 5 to 10% of the seeds are infected, our staff, they put it in front of X-ray and they try to remove the infected seed and salvage the seed, so that again and again our breeders don't have to submit the seeds to us. Now, normally this is the flow of germplasm. Accession number is given after viability testing is done, but many times our friends, they request that since that notification meeting is going to come because after the variety is recommended for release, many of our breeder friends, they don't submit the seed for 6 months to NBPGR for IC number allocation. But when next week that CVRC meeting is there, everybody will submit the seed and they want the IC number immediately. So I will request that because complete processing takes around 1 to 2 months, it is requested that everybody should in advance submit the seed. But still, in case of emergency, we at this stage, we issue the IC numbers after, and then viability testing is done. In case any of the variety which fails in viability testing, we approach the breeders again to resubmit the seeds. This is the what I was calling identification, copy of identification proposal, which is being submitted. So every crop has their own almost the same guidelines is there. So there is nothing which new I am showing. All the breeders will be having this pro forma, which they will be submitting to their respective workshops for release of the variety. So we need only one filled copy of this pro forma. And this is how we issue the certificate for release variety. Like this is to acknowledge that seed receipt of material Urdubin variety from Dr. Such and Such from Gujarat has been received in part of requirement for notification. And the material has been assigned national identity number, this thing. So this is the form of certificate which is being issued, which helps in, and copy is also again sent to DAC to facilitate the notification of the variety. In case of really hybrids, we issue IC numbers not only to the hybrid but also to all the parental lines which are involved in production of hybrid. In case many times what happens, in centrally released variety, okay, there is workshop. But in case of state release variety, many times workshop, they don't take place. They have to submit to SVRC. And for that, they need IC number. So this is the format which this is to acknowledge that receipt of seed of improved cultivar. We don't use the word release variety here, and we issue IC number. Then we very clearly mention that IC number is being issued only for the purpose of submission of identification proposal to SVRC, and this certificate should not be used for notification. After the variety is recommended by SVRC, they submit the proceeding of SVRC, then we issue the proper certificate which can be used for the notification of the variety. Then in case of vegetatively propagated crops, this is the type of certificate. So almost everything is okay, but here we also mention that in which nags it has been conserved, because as I told, vegetatively propagated crops, the seed has to be submitted in the national active germplasm sites. Like here you see, the breeder is PK Nayak from Sugarcane Research Station, Odisha. But he has submitted the material to SBI Coimbatore, which is the identified national active germplasm site in case of SBI Coimbatore. So in addition to the IC numbers, now in many crops, even the requirement for DNA fingerprinting has been made compulsory. So this is just for information of the participants that for getting DNA fingerprinting done from NBPDR, we would like separately. This is separate from what you have to submit to get IC numbers. This is the separate information which you have to submit, where you have to submit the copy of the proceedings with recommendations, then fresh and untreated seeds of varieties, hybrids, and parental lines in sufficient quantity. The quantity is 200 in case of self-pollinated crop, 500 in case of cross-pollinated crop. The fees is for 2,000 rupees for public sector and 6,000 rupees for private sector. Then submission of seeds of all parental line is mandatory in case of hybrid. And after some, because this takes a lot of time for germination, DNA isolation, PCR setup and electrophoresis, and therefore minimum 3 months have to be given to NBPDR, before which NBPDR can submit the report. And just briefly, around 6,600 release variety, they are conserved in National Gene Bank, which consists of different groups, that is cereals, millets, pseudo cereals, grain legumes, oilseeds, fiber, vegetables, fruits and nuts, medicinal and aromatic plants, spices, forage, and ornamentals. So with this, I would like to conclude my lecture. And if anybody has any question, I will be happy to answer. Thank you, madam.
Thank you, sir. In chat box, some questions are there, sir. So Can you see, sir? Or
Yeah, tamarind.
Tamarind.
Tamarind is, I think, for this will be tuber crop, means there is a spices institute there. What is that name in Kerala?
Yeah.
No, but tamarind will come under
Fruit plants, sir.
Fruit plants.
Yes, sir. And our headquarters, CIS Bikaner Regional Station, sir, Godhra, maintaining the germplasm at Chase Godhra, sir. So for this, sir, they can submit the graftings 10 to 30.
Okay, so now I remember actually for tamarind, I think in that we have not notified any this thing. 1 second.
Yes, sir. We also have released one variety, sir, Goma Pratik. At our
Now
station.
I have just checked. Actually, exclusively there is no center which has been identified as NAGs for tamarind. So that is why I told that we need to revisit the NAGs because new crops have come, new requirements have come, and like earlier we had 3 centers in millets. For sorghum we had Hyderabad. For small millets, we had Bangalore. For pearl millet, we had this Jodhpur as 3 NAGs. But now all the 3 have been incorporated in Indian Institute of Millet Research. So we have to re-notify the things. So these type of crops will be properly notified in the next stage of NAGs notification. I think this was one crop which has. There was no other question. So, Madam.
Yeah, if dear participants, if anybody has some question, so you can directly ask also. Because sir has given a very detailed account of millet genetic resources. Because sir is the curator of millet genetic resources in the National Gene Bank, and he is looking after all the important crops in the today's era. Because in the health time of health crisis, this millet crops play a very important role. And this year being the International Year of Millet, so certainly our millet crop is very. And sir has also announced that field day. I earlier announced to them also, because some were interested in sorghum and other millet crops. So I earlier also told. Date I was not knowing, but I told him means.
So today only we notified it. So
okay, okay. So 13th March, no?
Yeah.
Okay.
Yeah.
So means, yes, certainly they will attend crops and.
Hello. Hello, sir.
Please tell.
Yes, sir. Mulberry fruit, that is a minor fruit also. There any NAC center is.
Yeah, mulberry. Mulberry, it is there in Hosur, I think.
No, sir. Mulberry is there. We are conserving this large Vasur. I am from Vasur only, but it is mulberry fruits are categorized under the minor fruit, no, sir. For that, any minor fruit NAC center is there for
mulberry. This is the CSGRC Hosur.
Yes, sir. Yes, sir.
Yes, that's what so this is being this is already notified as NAGs in case of mulberry, and this is the only center. We have not notified any other. So now when you are saying as minor fruit, normally we don't notify NAGs as minor fruit like that. Maybe as temperate fruit we can notify. As tropical fruit, we can notify the center, not as a minor fruit center.
Okay.
You got my point?
Yes, sir. Yes, sir. I got it, sir.
So maybe we are working, and I think I was just yesterday our director sir was talking to somebody that within next 6 months we will finalize and revisit the NAGs, and all crop institutes and state agriculture universities will be expected to finalize this NAG center.
Okay, thank you, sir.
Yeah.
Okay, thank you, sir.
Sir, 1 or 2 more questions.
Yeah,
in chat box.
Is there any reason that Application for, yeah, application for IC number, as a habit, we don't reject, you know. I told you that our people, they work for hours to salvage the, although even if one infected seed, if we get, we can reject it. But we look it in microscope, X-ray, and try to remove the infested seed. So, but if more than 50% infestation is there, can you imagine from 2000 seed removing 1000 seed, that too through X-ray? So when there is lot of infestation is there, insect damage is there, other type of fungal damage is there, then the seeds are otherwise healthy seeds. If they are there, we don't reject. And also many times information is not given, that is the proposal, copy of identification proposal, or the workshop proceeding. We try to reach the breeders through phone and email, so that they submit it and we quickly give their IC numbers to them. So it is only because non-submission of any relevant material or information from the breeder, then only IC number is not given. Otherwise, we are not here to reject. We are here to facilitate the process of notification. We are not policing authority. It is our duty to process and conserve the seed and issue IC number, so that it is the notification is facilitated. So I hope this will answer. The next question is whether seed behavior like orthodox or recalcitrant seed is included in the this release proposal for different crops, different like for rice only. Rice Research Institute, that particular AICRP will be dealing. So similarly, in case of the different vegetatively propagated crops, you have different AICRPs. So all the norms will be as per your particular crop, whether it is propagated through seed or through propagating material. So it is not that in release proposal separately for, or this release proposal don't have any column for orthodox or recalcitrancy. Okay, so anything else?
So anybody can ask if they have any doubt there. And if not now, they can ask to sir through mail also. We'll be circulating mail and phone number also. So they can ask their doubt in future also. So, so if nothing is there, then I would like to thank sir. So sir, we thank you very much, sir, on behalf of training organizing committee, on behalf of participants. So we thank you very much for delivering such an important and excellent topic, sir. And
thank you so much, madam.
And clearing all the dots and good interaction with the participants.
Thank you. Thank you. Best wishes to you and all the participants. And again, once again, I invite you all to the Sorghum Germplasm Field Day on 13th March. We will send the letters to different crop institutes and vice chancellors. Thank you. Okay, madam. Then if you permit, I will leave the meeting.
Okay, sir. Okay,
thank
sure,
you,
sir. Thank you, sir. Okay, so dear participants, so today our next lecture is postponed at the last moment. Our resource person got some urgent meeting, so today we don't have any lecture now. So we will break for now for today's session, and tomorrow again session will start at 9:30 a.m. in the morning, and we will update about the tomorrow's schedule. Okay, so so everybody should join tomorrow. Tomorrow is also very good lecture. Okay, Mandalji, so you can close the meeting for today.